everyone. If you are watching this right now, you are watching the live recording of the TWIS podcast. Anything you see over the next 2 hours may or may not be included in the final podcast. So you'll have to just watch if you want to watch everything, or subscribe to the podcast if you would like the pretty edited version for your ears. Are we ready for a show? I think I've gotten the cue that we are live and people see us. So let us start the show in a 3, a 2, and this is TWIS. This Week in Science, episode number 785, recorded on Wednesday, August 5th, 2020. Why are lemurs? Hi, everyone. I'm Dr. Kiki, and tonight we will fill your head with happier cows, spinny sperm, and toxic webs. But first...
Disclaimer. The following program might be considered politically biased because it is almost entirely science-based. We have not taken time to include the other side of the arguments. We will to the world is round, knowing full well that there's still a robust argument amongst a few individuals somewhere around the world who have a different view of how the universe works. We will talk as if global warming is real and man-made and getting worse, despite the politicians with ties to the fossil fuel industry not being entirely convinced. We will talk about extinct life forms, ancient DNA, and evolution, without explaining that most of the world still entertains cartoonish children's stories as their origin. And we will talk about the very real threat from a pandemic virus. We will not allow anyone here to call it a hoax, not when there is now 1 American death every 80 seconds from COVID-19. Wear a mask, wash your hands, stay home, or better yet, head for the hills and don't ever plan on coming back. Whatever you decide, remember that if we all just took the drastic step of doing nothing together for just 3 weeks, we would be done with it. Instead, we're just beginning another episode of This Week in Science, coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We're back. We're here. We have the science. We do. And we have a scientist who's joined us for the evening. It's going to be very exciting. Tonight on the show, I have stories about spinny sperms and also new natural protection for multiple sclerosis and limits to our brain. Tonight we're going to be joined by Dr. Lydia Greene, who will be talking to us about lemurs. It's going to be very thrilling. Justin, what did you bring?
Interesting. I've got the body of an athlete, just not sure what sport it's suited to yet. Juno discovering activity on Jupiter and better mouse memories.
You have better mouse memories?
Yeah.
Better than bitter mouse memories. Blair, what's in the animal
I
corner?
have those too. I actually have a few of those too.
I have cows. I have frog vents. And I also have bad pandas.
Frog events?
Are there ever any good pandas in Blair's animal corner?
I think if we took a look back through the past, probably not.
No, it's always bad pandas.
Yeah.
I think we should have a TWIS sticker or T-shirt or something. I think you need to make some art, Blair. The bad panda art.
Is that like bad wolf?
Yeah.
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 That'll be good.
Start writing it everywhere.
It could be good. Oh, as we jump into the show, I would like to remind all of you that subscribing to TWIS will bring you TWIS every single week, right? It's a subscription to science-y goodness. You can find us on YouTube, Facebook, or just about any place podcasts are found. Look for This Week in Science or head to our website, twis.org. It's now time for the quick science stories. Let me start us off with some Martian ice sheets. I've got so much sibilance right there. That's kind of crazy. Ice sheets from Mars. Yes. Using an algorithm taking into account erosion processes, researchers at the University of British Columbia and their collaborators analyzed over 10,000 Martian valleys and compared them to subglacial channels in the Canadian Arctic archipelago. They found similarities that led them to a new hypothesis. That no, there never were any free-flowing rivers on Mars. None of that watery, rivery surface. No, these river-like channels that are seen on the surface of Mars today were formed by meltwaters beneath ice sheets, like those that used to cover much of the Earth. The idea would be even more supportive of life on Mars, as thick ice sheets would protect microbial life from radiation.
From radiation,
Yeah, and provide a kind of constant environment for them to live in. So ice sheets, that's the new idea now. What say you? Are you sad to see the
I like
rivers
this
go?
idea. No, well, because then what you have, is, especially under a melting glacial thing, is you have ponds, you have subglacial waterways, you have actually kind of a very robust, and then as you just pointed out in the beginning there, which the protection from radiation, which would have been crucial for Earth-like life to have survived there, would also be helpful.
Yeah, because Mars has never been known for having...
That's all good news for potential life on Mars.
Yeah, Mars has never really been known for having a big magnetic field to protect anything on its surface from solar radiation. Earth has a lovely magnetic field, which we say thank you to every day. Thank you for protecting me, magnetic field. I mean, I do, don't you? Justin, tell me about that athletic potential of yours. Will you be leaping across the surface of Mars?
Well, so this is the thing. Like, I've always felt like I am an athlete. I just never found a sport that I'm good at, but there may be help on the way. This is a new paper by University of South Australia sports scientist Professor Grant Tompkinson. He analyzes how a new portable, kind of cheap, I think it's like $8,000, 3D whole body scanner can spot athletic talents. Also monitoring changes in bodies over time. So this is a field called anthropometry. I'm not sure where to put the emphasis. It has been used for a long time to identify talent. It's used for this very thing, improve performance in athletes and check their health and that sort of thing. But it's usually done with different kinds of scans, even x-rays. With this, according to Professor Tompkinson, 3D scanning is less invasive than manual tests. And because it is fast, large samples can be easily measured. There's no need for physical contact. It doesn't require a lot of expertise to run the machinery. And it can measure body surface areas and volumes. Unlike x-rays, it doesn't, of course, emit any harmful radiation. For this particular test, there was 49 athletes, 30 women and 19 men. They subjected them to a series of 35-second long scans and got millions of measurements within 2 millimeters of accuracy about their different body configurations. And so what they're doing is they're basically finding people who are already really good athletes in their fields and sort of figuring out what are the measurements that make a good long distance runner versus somebody who can lift a lot of weight or whatever their athletic skill might already be.
But
But
that doesn't take into account muscle fibers and the fast twitch versus slow twitch fibers and what proportion of the fibers in the what?
So if you think about it, I mean, and this was a lot of like sort of what I've looked at this too was like, well, how much information are you actually getting out of there? They're looking for small things. They're looking for asymmetries. Is something as big as scoliosis, one leg being longer than the other, arm lengths overall in terms of giving reach. But
Don't
then
tell me that my
density,
crooked spine can't do a back bend. No.
no. Just maybe not in the Olympics. Sorry.
Right.
Okay.
Sorry.
Sorry. Didn't mean to break that bubble. But I would think that you could tell perhaps that the different twitchy muscles based on how they lay upon the leg. I mean, that sort of bulky calf muscle is probably more of a lifter's muscle than a long distance runner's muscle, or a sprinter's muscle more than a long distance muscle. So you may be able to sort of see that because especially they say marathon runners have very defined physiques, shorter, lighter, longer legs relative to their torso. And then they're saying they can also track changes in whole body and lean mass using this scanning. I mean, you're not going to replace the combine event in the NFL where they're putting people through the leaping and the running, who's fast, and it's not really going to replace any of that. I could sort of see it being used as a training skill, but I do like the idea that you could use it to tell what type of athlete body you most resemble. So you could be like, oh, I might get a little printout. It's like, oh, I should invest some time playing badminton because apparently I have a badminton athlete's body. I didn't know, haven't been playing hard enough. I should go. Now that's what I should go try doing because maybe I'm better suited to it.
This is also your excuse to not work out, right?
Exactly.
Like, I'd go for a run, but I did a 3D body scan. It turns out I'm just not a runner.
Just not a runner. Yeah. Yeah.
It's reminding me very much of those career aptitude tests that are given to you in high school, and they tell you what you're going to be. Based on your skill in high school, your knowledge in high school, you're going to be a secretary. You're going to be, an astronaut. None of the options on that test ended up being science communicator.
Yeah.
Okay.
None of those options were available.
Can I tell you what my top two were? I remember this test because it led to a summer job. My top two were scientist and actor.
That's great. My two were park ranger or chef.
You're right. Oh, you feed animals.
You're right in there too. Okay, so it was accurate for you, not for me. I was never very good at tests.
Oh. Well, you're not supposed to be good at the test. Kiki. You're just supposed to be honest.
Yeah.
What did it tell you, Kiki?
I don't even remember. I looked at
Martial artist.
Threw it. I was like, that says nothing about me. I think it said you should go in the army. And I was like, no.
Oh.
No.
Just because I'm violent doesn't mean I want. It's totally different. I just want to throw inanimate objects across a room and scream.
I just want to kick Justin's shins. No.
You could see that on the body scan.
Right.
Oh, that's
you could.
true.
Yeah. You can see the lumps on my shins. According to our test, you work with somebody who has a temper. Yes.
I don't know. It'll be interesting to see where this goes, whether it's more than, so many of these kind of, IQ tests, these very I don't know. They're trying to do very specific stuff, but is it really going to tell potential, or will it be more used as a tool for refinement?
As a slight disclaimer or caveat to this, I have seen a lot of commercials for some sort of like, instead of having a healthcare system, just walk into this body scan and it tells you everything you need to know. No, it won't. It's not good like that yet. It's not taking any blood samples. It's running a little bit of electricity to your body and going, oh, it's testing your heart rate. Like, that's fine. But that's not really replacing your doctor anytime soon. But the timing of this coming out and that being popularized or advertised kind of makes it a little bit suspicious that this is coming out right now.
Science by press release.
Yeah. Science by press release a little bit. Yeah, maybe.
But
There's no
fun
results to
technology.
this. Didn't say like it's like, hey,
Maybe
we can look at.
Maybe it'll just help, our genes fit better in the future. I don't know.
No, but that's
Blair?
actually probably one of the highest best uses. Yeah. No, this is absolutely true. A hip, leg, butt scan before you like buy a pair
and
of
then 3D
jeans.
print your jeans. There you go.
Yeah, well, yeah, or just order off the
Clothes
rack. But
that really
having them
are
designed-
skin tight.
Yeah.
No, this for tailors. This is like probably and then you walk into the body scan, you have that recorded. Depending on how much you fluctuate, you go back with more or less frequency, and then you can order your clothes online, custom fit, because they've all been tailored to you. That's probably the best use of this technology.
Yeah, I think so. Could make us happier in the future shopping, but Blair, what's going to make cows happier?
Well, I don't know, but I do have a study related to how we can tell if they're happy or not. So we know that cows actually are social creatures. A lot of people don't realize that, but people who study cows certainly know that. And that they have complex social relationships that change and the group dynamics constantly evolve. So a team of Chilean and US scientists spent 30 days observing a small herd of dairy cows that had recently given birth to understand a particular part of their bovine interactions, which is social grooming behavior, also known as allo grooming. And the reason this is kind of so important to look at is because you don't just throw a bunch of cows together on a field and call it a day. In dairy production systems, they're constantly shuffled into different groups depending on their lactation stage, nutrition requirements, where they are in breeding, etc. And every time they do that, they have to reestablish their social structure. So knowing how to kind of read their behavior, know if they're adapting to their new social structure well or not is really important, because a happier cow is a healthier cow, and a healthier cow definitely makes better food products and will live longer and will have more babies, et cetera, et cetera. So by looking at this, they looked at over 1,300 allo grooming events from 38 cows during this month-long experiment, and they found a lot of different behaviors and patterns of those behaviors. They looked at things like animal age and social rank, and they found, for example, that cows tend to groom individuals that had previously groomed them, stuff we've talked about with other animals, so a mutual cooperation. But they also tended to prefer individuals of a similar age, suggesting familiarity from growing up together or just recognizing their similar age. I don't know, some other reason. And then they also found that older individuals groomed more cows than younger ones. So allo grooming could be also related to seniority in some way. So all of this to say, they could actually look at how the grooming was happening, how often it was happening, and who was doing the grooming to kind of draw conclusions about if the group was acclimating well after a kind of a remixing or not, and move forward therefore. So they know that this is something that could be used in the future for farmers to monitor the social aspects of cows and the emotional relationships within their group. And if they are able to see that cows are grooming each other, that means that they are getting along. But if grooming declines, that could be a sign of impaired welfare. And that is a great opportunity to check, make sure nobody's sick, nobody's not getting along, maybe check your stress hormones. There's lots of things you could do, right? Once you kind of recognize that there's this trigger. So I just wanted to bring this story because I think it's important for us to think where our food comes from. If you're a vegetarian or not, you might still drink milk. If you're a vegan, you might, I don't know, still wear leather or use other products that benefit from livestock in some way. And it's important for us to understand their social structures because that can help us take better care of them. Kiki, you're muted.
I didn't know that cows engaged in this social grooming behavior.
Yeah,
I've never
there you go.
noticed it. And
And
there's
that
cows
is
all
the reason
around.
I brought the story. I think it's a lot of
Yeah.
people don't know how complex cows' social structure is.
I'll have to watch
Just
it a little
drive by them
closer.
on Highway 5 and see them sitting around. Think that's it.
Moo.
Yeah.
You just think of the moo cows, but you don't think about them as individuals, as a group, how they get along with each other.
That's right.
They're social animals, so it's not.
But yeah, I have noticed that they do cluster. Even if they have like a ton of room to go wander around, they will all sort of hang out together in whatever patch of
if they like each
pasture
other.
they're in. But I guess my local cows are very, very well adapted to each other because
I think Davis cows are on the whole they're moo cows. They're happy cows.
They're pretty happy. I mean, they really like to the point where I've also sometimes like spread out. What are you doing? There's like all of this acreage you have here, and they're all like clustered up right next to each other. Like some of them are like pinned in. Like even if they wanted to go for a walk, they're not going anywhere. They're stuck.
Speaking of happiness, let's talk about sperm.
Always.
When you think of sperm, you think of that little flapping flagella going from side to side, swimming the sperm toward their final destination, death or egg. Apparently,
It's almost always death.
Yeah, mostly death, yes. But apparently, sperm don't really swim. We've had the wrong idea for basically as long as we've had microscopes. So
stop. What?
Yes, they spin.
No.
Sperm spin.
No.
Yes.
What?
So there's a new investigation, of sperm motility, motion of sperm, determined that what we saw as this side-to-side motion was actually an optical illusion because we were looking at sperm as a 2D image through a microscope.
It's like a corkscrew into a bottle of wine. Is that what you're saying?
Yes. So instead of flapping back and forth, what happens is the head of the sperm spins like a top. And then to compensate for the spinning of the top, the flagella actually has a lopsided movement it's asymmetrical. It only moves to one side. But because the sperm body is spinning, the movement of the flagella appears to be going back and forth.
So
but
do they all spin in the same direction?
Oh.
Yes, I think they do. But I think that when they don't spin in the same direction, they don't do as well. I don't know. Actually, this is a really interesting question.
Does it depend on the hemisphere?
I do not know. I think, that's a really great question.
Or do all animals have the same spinning sperm?
I think that the hemisphere just
also.
Yeah.
I thought the hemisphere came down to who designed the toilets.
Yeah, I think that's true.
Yeah.
I was making a bad joke.
Yeah. But no,
which I
always welcome.
Yes.
Yes.
So wait, now I'm confused at the propulsion. Like what's moving the spinning if it's not the flagella thing?
But the flagella is moving.
It's part of that propulsion. It's just cork.
Yes.
So it's like a corkscrew. It's like they're like corkscrewing through the fluid, right? Is that...
Yes. It's like a spinning top, like a corkscrew.
It's kind of viscous fluid, so it's like
So they drill. If you could imagine, if you've ever used a drill that has the handle on it that you spin around the drill bit, that's kind of the way I think of it and how they're moving. But they move in 3-dimensional space as opposed to just this 2-dimensional back and forth movement. And they have this, and it's all rotational.
Today will go down in history as the day I learned that sperm spin. I'm never going to forget it. It's great.
I think this understanding could affect so much understanding of sperm motility and issues with infertility. Yeah, sperm competition. And like Lydia brought up, if there are sperm that swim in different directions, what does that mean? How are the molecules set up within the sperm for the molecular motor to move, and how does that affect how they perform?
And also how they fertilize.
And how they fertilize,
How they fertilize, and now it makes me picture them like how the NASCAR cars all get really close to each other for drag. Like if they are like not agreeing on a direction, do they not make it as far? That little group, like, are they working against each other, sending little turbulences if they're not spinning the same way? If they're not working as a team initially, at least.
Like ducks flying, and like the one in the lead has to like go to the back.
Yeah. Yeah.
Yeah, it's fascinating. And it really, you're right, Justin. What does it mean for turbulence? And how do they deal with that? This has so many layers of complexity now that I never ever considered. Sperm, so complex. They spin. All right, tell me about Jupiter, Justin.
Oh, yeah. So there's activity on Jupiter that we weren't aware of before. This is NASA's Jupiter orbiting spacecraft Juno, sparking new questions about the gas giant as it unexpectedly discovered lightning in the planet's upper atmosphere. This is published in August 5th, which is, I guess, today. Journal Nature. Jupiter's gaseous atmosphere seems placid from the distance, but up close, clouds roil in a turbulent, chemically dynamic realm, according to this research paper. This is who am I quoting? Jonathan Lunin, professor of physical sciences and chair of the Department of Astronomy at Cornell University. On the night side of Jupiter, you see fairly frequent flashes, as if you were above an active thunderstorm on Earth. You get these tall columns and anvils of clouds, and the lightning is going continuously. We can get some pretty substantial lightning here on Earth, and the same is true for Jupiter. So previously, Voyager 1, Galileo, New Horizons had all observed lightning of some form going on Jupiter, but they couldn't really tell. It looked like it was coming from deep down inside. Juno has a camera that is designed to detect much dimmer sources of light, and it's also just sort of more able to tightly control its focus, as opposed to its resolution and sort of depth of field than the others. And discovered lightning where we actually didn't expect it to be possible. And what they think is happening is that there's ammonia that is in the upper atmosphere. There's water and other elements there as well, but they would normally freeze at this far out from this core of this gas giant. They would be expected to be frozen, but if there's enough ammonia there, it keeps the freezing from taking place, and therefore you can continue to have this electric activity taking place. So cool, something we didn't know. This is also Yuri Aglamov, who is a postdoctoral candidate at Cornell, saying shallow lightning hadn't really been expected and indicates that there's an unexpected process causing it. It's one more way in which Juno's observations show a much more complex atmosphere of Jupiter than had been predicted. We know enough now to ask the right questions about the processes going on there, but as Juno shows, we're in a stage where every answer also tends to multiply the questions.
Juno nothing. Sorry. I couldn't help myself. From what I think is amazing is we've seen Jupiter. We've had images of Jupiter, the Great Red Spot, all these atmospheric clouds for so long, had ideas about what's going on. But now that we're actually there and we are, we've got Juno spinning around it and making these passes and getting in and looking at different areas of the atmosphere and really digging into it, we're starting to find out so much more. And it's amazing that Juno is still going. It was supposed to be destroyed by radiation and crushed by magnetic fields long ago. So this is pretty cool. Lightning. It's awesome. Very cool. All right, last quick story. Orb weaver spider webs might contain neurotoxins.
Of course.
Of course.
Already the most terrifying spiders, in my opinion. Might as well throw that on there.
I love orb spiders. The way the dew glistens off their beautiful orbish web.
The way they build a new spider web every morning, so it's extra see-through, so you walk right into it. The fact that they love to make them right at face level.
Yes.
It's all great.
This story specifically spoke to me today, as last night, as I was pulling toilet paper off of a roll, a spider scooted out from between the sheets of toilet paper in my toilet paper roll, and anyway.
Hi, bud. No thanks.
that toilet paper roll.
Yeah, not exactly where you would expect.
No, but anyway, yes. You walk through a spider web and you're like, no big deal. But maybe it's just because you're really big. This week, a reporter, Christy Wilcox, reports on a paper that reports neurotoxin-like compounds in the silk glands of banana spiders. And these neurotoxin-like compounds might allow them to weave webs of doom for their prey. There are many tales of researchers who are interested in these spiders or the general class of orb spiders and how their prey, even before being bitten by the spider, tend to show some listlessness or symptoms of what you would expect from neurotoxin poisoning. And that's what led to this direction of inquiry, are these kind of symptoms that the prey species were exhibiting. And they've found that in other species' nests, when they've rinsed the webs and then exposed prey species to the stuff that comes off of the webs in solution, that it's caused neurotoxic effects. So we think that maybe it's a general thing that all orb spiders might be capable of putting neurotoxins in their webs. And wouldn't that be helpful if you've got a prey item that's across the web from you?
Yeah, you don't want them struggling
Slow it
so
down.
hard they might pop
Slow
off.
it down.
Yeah.
Or they could even be dangerous to you. So some considerations there when you next walk through an orb spider's web.
Or the next time you rewatch Lord of the Rings.
Oh, yeah.
Oh, dear. Yes.
Oh, good point.
Very good point. If you just tuned in, you are listening to This Week in Science. If you're interested in a TWIS t-shirt or mug or other item of TWIS merchandise, head on over to twis.org and click on our Zazzle link to browse our store and support TWIS.
We have face masks, too.
Yeah, we do have face masks. Faux show. They are our most popular item right now,
Yes.
which makes me so happy. Thank you,
Way to
everyone,
meet demand. People
for
are being
protecting
safe
people.
and buying our stuff.
Yes. All right, time for the interview. Tonight we are joined by Dr. Lydia Greene. Dr. Greene is an NSF postdoctoral fellow in biology in the Nicholas School of the Environment at Duke University, working at the Duke Lemur Center, studying the ecology of Madagascar's lemurs by looking at their poop. Welcome to the show. Thank you for
Thanks for
joining
having
us.
me.
It's great to get to talk to you. I came across you on Instagram and your Instagram account at Lemur Scientist, and I was just smitten by all the lemur photos that you
They're
post.
amazing. The animals are amazing. The photos don't do them justice, but we try as much as we can to highlight them. Yeah, but they're
So-
amazing.
Do you ask them to pose for you, or is this no?
So the lemurs that we have here at the Duke Lemur Center are so used to having people around them, and so they don't mind at all. They're like quite hams for the camera. The lemurs in Madagascar are much harder to photograph because they're so much less habituated to people, and so for those guys, it's usually a long lens and very blurry and millions of horrible photos before you can find one that you can zoom in on. So it's a real treat to see them up close at the Lemur Center.
How did you get interested in lemurs? What led to your interest?
Serendipity, I think. I came to Duke as an undergrad after a sort of failed attempt at a professional ballet career. And I came to Duke for college just because it's where my mother thought I should apply. And I got in sort of miraculously, I think. And I needed a work study job. And I went to the work study job fair. And there was like this table with this sort of life-size stuffed shafak, which is a type of lemur on it, a stuffed animal that was like a my size. I was like, what is that? And they said, we're recruiting tour guides for the Duke Lemur Center. And I was like, the Duke Lemming Center? And they were like, no, Duke has lemurs. We have a center for lemurs. And I was like, tell me more. And so they were recruiting tour guides. And within my first week of college, I basically signed up as a work study tour guide. And from there, it just kind of snowballed. So I started taking classes in anthropology. I joined a lemur-focused research lab. I sort of began my own research projects. And it's just one of those things where you fall in love with these animals so quickly. And most of us never turn back into anything else again. So yeah, it's sort of a black hole you fall into, a great black hole.
Wonderful. It sounds like it wouldn't be one you'd want to escape from.
No.
When I think of how most people probably have interacted with lemurs, it's maybe at zoos or through nature documentaries. And specifically in nature documentaries, we see lots of behaviors that people like David Attenborough tell us about and like make very special. But what about them fascinates you the most?
So I think everyone's drawn at first to just the look of the animals, but when you sort of get steeped in lemur biology, the diversity of species, I think, is what really starts to become fascinating, and the origin by which that diversity arose. So I'll take like a few minutes just to sort of get the basics of lemur sort of biogeography going. But if you picture it 50 to 60 million years ago on Earth, when the dinosaurs start to go extinct or totally go extinct and mammals start to arise and primates start to evolve in mainland Africa, Madagascar had already been an island for like 30 million years. And so one of the biggest questions in lemur biology had always been, how did lemurs get, or early primates get, from mainland Africa all the way over to Madagascar? And it turns out that the evidence points to a sort of early primate ancestor rafting across the Mozambique Channel from mainland Africa to Madagascar, a small colony of sort of pre-lemurs. And when they got to Madagascar, the small colony of lemurs stepped upon an island where there was a lot of open habitats, diverse habitats. There were no mammalian competition, no mammalian predators. And so these animals just sort of exploded in adaptive radiation. And so from a small founding event to the 108 species that are extant on Madagascar today, as well as a bunch of species that have gone extinct. And so we just get this sort of enormous diversity of species that arose from this sort of crazy serendipitous event of lemurs getting to Madagascar in the first place. So when you start to sort of look at that story and unravel the sort of way by which these creatures came to be, you become all the more fascinated by the myriad different directions that the species sort of radiated into, and how they carved out lives for themselves. Sort of a ramble.
Kiki, you're muted again.
It's because I go in the back and I try and find images, and then I come and I mute myself so that there's no tick-tick-tick-tick-tacking on the keyboard. But I am fascinated by that. I guess the way that the movement of Madagascar geologically, how that converged with the movement of the species across oceans, and then how it also created the ecosystems, the environments in Madagascar, which led to that diversification, and how you're looking at time, geology, ecology, like all these pieces falling into place.
Yeah, we say Madagascar is sort of one of the greatest natural experiments that sort of Mother Nature has given us. So during Pangea, Madagascar is the sort of sandwiched between India and Africa, and it's also attached to Antarctica a little bit. And as Pangea starts to break up, Madagascar splits off first from mainland Africa, and then is attached to India for a while during the beginning of the journey across what is now called the Indian Ocean. And so Madagascar then separates from India about 80 to 90 million years ago. And at that point, Madagascar was quite far south. And so for the past sort of 80 to 90 million years, it's been isolated but drifting farther north. And that drift has also had, oh, look, you've got a perfect map of it. And so that northward migration also has big implications for the types of habitats that have been on Madagascar as lemurs arrived, and then also as the other mammals arrived, because we think there were 3 other rafting events that brought over the rodents, the tenrecs, and the carnivores as well. So you're exactly right. This interplay of geology, of ecology, and of time becomes really critical in piecing together the story of Madagascar's mammals.
They didn't all come over on one big raft?
No, we think that there were 4 rafting events. Genetic dating really points to there being four distinct rafting events, which is kind of crazy as well. So you can imagine lemurs get there first, and they were there for, a number of million years before the carnivores showed up. And you can imagine the lemur, the first time it laid eyes on this carnivore that had just arrived, fresh on shore, and was like chasing it around, and it had never experienced anything like this in its history.
Oh my gosh.
And then there's also, it's got to be for you said 108 different species that are there now. So
yeah, a few days ago.
One of the things about Madagascar is, right, it's not just an island of a forest of one particular type. It's for just a kind of a somewhat small body mass. It's actually got kind of a diverse set of biomes to it.
Yeah, so Madagascar is famous for having a real diversity of landscapes. And so you've got like rainforest in the east, you've got a central high plateau that has endemic grasslands. You also have western dry deciduous forest, and then you have a southern spiny desert. But then within each of those ecosystems, you also have sort of microhabitats and you have pockets of microendemism. So even within the rainforest, you don't get the same assemblages of species in the southern tip that you would get in the north. So you have this crazy, crazy microendemism. And I guess the mouse lemurs are one of the best examples of this, that these tiny little primates, we now recognize 25 species all over the island of Madagascar. And so each one has its own little habitat that it lives in. So yeah, it's mind-blowing actually how much microendemism, and the lemurs are an example of that, but a lot of the herps and the birds and the micro mammals also show this kind of crazy speciation.
And it's just because there's so many diverse, the ecosystem is so rich. There's so many different, it's like somebody chopped up Madagascar and said, be everything.
Yeah, and there's a number of theories to explain sort of why you get these pockets of endemism, and the one that I'm most familiar with has to do with watersheds and rivers. And so in some of these areas, you get almost sort of like parallel rivers, and different groups of species will live within the boundaries of the rivers. And so the rivers actually provide the natural boundary that differentiates species, which is kind of cool. So you just cross the river and you might find different lemur species all of a sudden.
That's amazing. So that gets at there's the idea in speciation of when is it actually a distinct species as opposed to a subspecies, and when can crossbreeding take place? Is it reproductively isolated, or is it geographically isolated? Do you know for the species in Madagascar how does that? There are diurnal daytime lemurs and nighttime lemurs, so those are going to be separated by time. But what is there separating the lemurs? Do you know?
Well, I will also point out that there are cathemeral lemurs as well. So that's an activity pattern. Cathemeral. So that's an activity pattern.
I haven't heard this one.
Yeah, so you can be awake basically at any time. And so we see this in a lot of the species of brown lemur, like the blue-eyed blacks and other ones that you can find at a lot of zoos around. And so what this means is they can be awake they can either do nocturnal or diurnal lives, and they can change throughout the year. And so often what you'll see is when it's really, really hot, they'll choose to be awake at night when it's cooler to forage and doesn't waste as much energy. And then when it's cooler, they'll choose to forage during the day. So you get this seasonal pattern to that activity, which is really, really cool. So yeah, you have diurnal, nocturnal, and cathemeral species. Yeah, Madagascar is crazy.
That's my word of the day, cathemeral.
But to the bigger question of maintaining sort of species diversity, that brings in sort of the concept of niche differentiation. So we do have 108 species. All 108 don't live in the same forest at the same time, but these 108 species basically stem from 15 groups, 15 genera. And so in many habitats within Madagascar, you'll have representatives from most of those genera, often living sympatrically, but you more rarely have multiple members from the same lineage living in sympatry. So the shafaks are a great example of that. There's 9 species of shafak, and no 2 species lives in the same habitat at the same time. So you have 3 different rainforest specialists, you have 4 different dry forest specialists, but none of them ever meet. So every forest you go to, you can sort of find a different assemblage of species, but you'll be able to recognize like, oh, that's a brown lemur, oh, that's a shafak, oh, that's a mouse lemur, but it might be a different species.
That is
That's
fascinating.
So at my zoo, in our lemur exhibit, we have 7 different species in one forest, and we have the black and white ruffs that hang out at the very tip top of the trees. And you have the ring-tailed lemurs that are like the ground lemurs; they're always down on the ground, and the crown lemurs are kind of in the middle. And so it seems like that's how it kind of works in Madagascar as well.
Right. So you have this temporal differentiation where you can sort of be diurnal, nocturnal, cathemeral. You have this sort of strata differentiation where you exactly right. You have rough lemurs up in the canopy and ring-tailed lemurs being more terrestrial, although those 2 species wouldn't ever meet in Madagascar. But then you also have differentiation by diet. So you've got leaf-eating lemurs, fruit-eating lemurs, lemurs that are more insectivorous, lemurs that are more specialized, more generalized. So you can differentiate by diet. So there's so many different ways that these animals differentiate within their habitats, and that's why these habitats can really support this diverse array of species.
And on that mention of diet, diet leads to poop. And so can you tell us a bit about what you're studying specifically and why you're looking at it?
Yeah, so I study the gut microbiome of lemurs, which means I do need to collect a lot of poop. And luckily, lemurs provide bountiful samples for me. So it's a nice research area because it's so productive. So basically what I'm really interested in is understanding how this gut microbiome relates to the diet of the animals and whether that diet is across species. So comparing a leaf eater to a fruit eater to an insectivore, but also comparing things like how within a species, seasonal change in environment will change the microbiome, how changes in habitat quality or landscape might be reflected in different microbiomes, and all those sorts of variables that are related to diet. So most of my work is basically following around lemurs with tubes and waiting for them to finish their morning coffee. And then collecting samples non-invasively. So all of my work is non-invasive and hands-off, which is really nice as well. And then for the microbiome, usually what I do is sequence a marker gene that is common to all bacteria and archaea. And that gene allows us to figure out which microbes live in which samples and at what relative abundance. And then we can piece back together which inhabitants were there and at what time. And that can really help us figure out how these microbes are adapting to different diets and within these different host systems. So,
what kind of microbial populations, like what is normal, or with the diversity of different diets, more insect eaters, plant eaters, frutivores?
Frugivore, yeah.
Frutivores, yes. I almost went fruitarian and I said, no, that's not right.
It works.
What kind of divergence do you see in the microbial gut populations? Is there a big difference?
Yeah, so what we're sort of piecing together in Madagascar is the same way we were talking about these assemblages of species, is that within a habitat, you'll have many representatives from different lineages. And it's become clear that the microbiome relates to your lineage. So a shafak living in a dry forest is going to have a microbiome that looks more like a shafak from a rainforest than it will from a brown lemur that lives in its habitat. So you have these clear patterns by the type of lemur, but then within these lineages, you have clear structuring by the type of habitat that they live in, by the diet that they're eating. So you have sort of multiple layers of different patterns that we could compare. And we even find fun things like for 4 lemur species living in the same habitat, so they're sympatric and they're all leaf eaters, but they eat slightly different leaf-based diets. We can see really, really clear signatures in their gut microbiomes that might help them survive on those sort of very, very different diets, but within this dietary category. So there's a lot to play with.
What are you going to do with the understanding of the microbiome? I mean, I can see understanding how to feed the captive lemurs better, but is there more beyond that?
Yeah, so a big one is understanding how we can help keep our animals healthy and well in captivity, and understanding diets and nutrition and things like that. I think from an evolutionary perspective, I'm really fascinated by understanding the sort of symbiotic relationship between animals and their microbes, and this idea that microbes allow us to do things that we couldn't do on our own. Microbes have this incredible wealth of metabolic capacity that we as eukaryotes don't have. And so there's a lot of functions that our microbes confer on us, and different animals end up getting different functions based on which microbes they have. And so microbes can enable leaf eating. They can enable living in particular areas or at particular times, or eating different diets. And so understanding how microbes underlie specialization, ability to adapt, I think that's a really, really fun place to be in science right now. And I do think it has conservation implications, because if we want to understand why certain species are more susceptible to habitat loss and climate change, microbes might be part of that answer, if microbes are really what are enabling animals to do certain things. So I think we sort of want to picture the whole... Sorry, go ahead.
Yeah. No, there might even be like a little bit of a biotech aspect to this too. And I remember it was a while ago, we had a story where it was I think 40 gut microbes that were the same. 40, maybe it was even 80, that were identical between red pandas, regular pandas, and a panda lemur, bamboo eating lemur. And it also,
that was my friend Aaron.
Some of them, and some of them were even like apparently in termites, which is also eating like a very rough. And so, you start looking at something like this, and you get this idea like, okay, well, if we ever need to break down bamboo as a biofuel or something like this, maybe you look to these creatures and how they've been doing it as a diet to find candidates for creating the enzymes in order to do it. But also, yeah, if we, as climate change tends to affect things, knowing, and if there's a plant that's not going to survive anymore, but you've got an idea of what would still grow and is closest to what their diet was before, that their microbiome can handle, for maybe a relative who's eating it somewhere else on the island already, that becomes huge for conservation as well.
Well, so to sort of think about Dr. Erin McKenney, who wrote the panda paper and the lemur paper that you're talking about, and sort of to invoke an Erin McKenney sense here, what Erin helped me think about when I was a graduate student, because she was a couple of years ahead of me, and we collaborated on some of these lemur projects, is if you think about it, gut microbes have been around since the evolution of a one-way gut. The evolution of the gut system itself went hand in hand with the evolution of the gut microbiome. We've never had a gastrointestinal system that doesn't have microbes in it. Those two came together. And so there are definitely microbes that are shared basically across all of us, because they were probably around when the gut first evolved and before we all diversified into the different species we see today. So there are definitely bugs in our microbiomes that are consistent across lemurs, across chimps, across termites. So things that pop up a lot like Prevotella, Bacteroides, and you see these microbes all over the literature and all over every sample. But then you do have these microbes that seem to be like species specific or diet specific, and those could be a really interesting place to play for looking at metabolic capacities that we don't yet know about. And again, this is where Madagascar, I think, becomes fascinating, because it turns out that some of these lemurs have actually quite a lot of like unassigned taxa in their samples. They don't match against online databases that are mostly built from human and model systems. And so some of these species of lemur, I blast their microbiome against these established online databases, and like 30% of the microbiome comes back as like, no idea what that is.
We don't know that does.
And so who knows what untapped potential is in these consortia in terms of what they're able to do.
Yeah.
That's a fascinating thing to think about.
Isolation and naming potential, right? Don't you get to name them all that's just fun. That's just a blast then, right?
Yeah, I don't see myself like culturing like trillions of bacteria for the rest of my career just for the naming rights, but yeah.
No, but just a couple of them, as Christmas presents.
Do lemurs? We know that in like koalas, they have a fecal transfer from the mother to the offspring. Is there anything like that in lemurs?
Yep, coprophagy. So that's the term for poop eating. And so we do see coprophagy, especially in infants, and usually from mom. So it's not that unusual to see an infant just go up to mom, and we do think that that's a way that they're potentially seeding their microbiome. In some lemurs, there's an idea of what's called cecotrophy. And so, so back up and give a little bit more context. So within the lemur gastrointestinal system, you have the intestines, but you also have this structure called a cecum. And in the human system, it's kind of what the appendix is, but in lemurs, this appendix is like fully blown out and really elaborate and complicated, and especially in the leaf eaters, because the cecum is the powerhouse site of microbial fermentation. So if you're a leaf eater, you basically shunt your leaves into your cecum where microbes attack it and turn it into nutrients for you. And so there's a suggestion in some of these leaf-eating species that they actually excrete the digestive from their cecum, and then they eat it again. And then by this point, the microbes have attacked it and have released all of these nutrients in it. And so by re-eating it, you are really, really quickly able to absorb all of those nutrients much faster than you could otherwise. And so there's not only coprophagy, there's also this idea of cecotrophy in some species.
So crazy question.
Yeah.
If you took one type of lemur as a baby and had it raised by another type of lemur, and then they ate that mom's poop,
what would happen?
That mean that they could then shift their diet?
No, I think is the big take home.
Okay.
I think if you had a leaf-eating lemur raised by a fruit eater and trying to eat the diet of a fruit eater, it would probably not survive very long. I think, the example shown up on the screen right now is a folivorous lemur. This is a leaf-eating lemur, and you look at the craziness of this gastrointestinal system, and you imagine trying to stuff a lot of fruit in that system that's going to ferment very quickly. This animal cannot just adapt to eating a fruit-based diet, and this is because feeding strategy, which is what evolution tells you should be eating, and your diet usually match, right? So evolution says you should be a leaf eater. You've got shearing teeth, you've got the gastrointestinal system and the microbiome to support leaf fermentation. But if you go on to just eating fruit, the system wasn't designed to support that. And so it's probably not going to adapt very well.
Interesting.
And I doubt we would ever get ethical permission to do that experiment anyway.
Well, so the only real reason I'm asking that question is that I know that, Madagascar is kind of a limited space. It's an island, and there's habitat destruction issues there. And so I'm just trying to imagine how you could push a population that lost its space into a new space that maybe required a different tactic or a different type of plant or something like that.
So I think that's a fun idea to think about. But what I would say, especially for these leaf eaters, is that they and their microbiomes are so specialized on the diets in their microhabitat that they've evolved to eat, that this is a pretty specialized and co-specialized system that you can't just uproot and move it somewhere else. And sort of a great example of that is the fact that most leaf-eating lemurs do not survive in captivity. Most of the lemurs that you're going to see under human care are your omnivores and your fruit eaters. And it's really, really challenging to keep these species, these leaf eaters, healthy under human care because they're so specialized on the specific diets that they eat. And so things like translocations, moving animals around, might be doable if you're moving them with sort of within the same habitat type, but you're not going to be able to transport a rainforest species into the dry forest and poof, that's probably not going to work.
How well do different types of lemurs get along?
With each other?
Yeah. Like with a different type, not within the group, but with other groups. Because I don't know anything about. I don't even know if they're more baboonish or bonobo-ish with themselves or each other. Like I don't know where they fall.
I don't do baboons.
Yeah, no, I would stay away. Yeah,
they're terrifying to me.
Yeah.
So there's sort of the question of how do lemurs respond when they meet other lemurs of their own species, but then how do lemurs respond when they meet lemurs of different species? And those are two completely different kettles of fish. So lemurs do have territories, and so they do maintain their territories. Some species are more territorial than others, but in general, when they come across a conspecific, a member of their own species that's sort of invading their territory, they're not going to behave very kindly to that. But we often have the case where species will live really, really close to members of other species and not care at all. And so actually at the Lemur Center here in North Carolina, we have large forested enclosures where we can put multiple social groups of different species into the same forested enclosure and they get along just fine. But wouldn't necessarily.
You put in another one that matches one that's in there, and then it's like mating competition problems all of a sudden. Now there's a reason to keep that group away from this group because they might steal my girlfriend lemur.
Right. And I won't go into there's a whole research area of animal friendships that I don't study at all and I don't know much about it, but I will say I have seen in Madagascar cases where you've got a brown lemur species and a shifox species that tend to hang out together. And so the guides will often say that the way they find the shifox is by first finding the red-bellied lemurs. And so I don't know if they hang out because it's the best tree and everybody wants the best tree, or if they're actually hanging out with each other for a particular reason, like predator avoidance or something like that. But yeah, you do sometimes see these guys hanging out together and it makes you feel like, oh, even if it's not what's really happening.
What have you enjoyed more in searching for lemur poop, going to Madagascar and searching in the wild, or having it easier in the Lemur Center, the Duke Lemur Center?
Yeah, there's definitely something really nice about working at the Lemur Center because you're going to get those samples. There's just no way you're not going to get that science done. And you can also sample them at a much more consistent and routine frequency than you could in Madagascar. I know I can watch Beatrice all day long and get every sample she excretes that day if I wanted to. So there's something really nice about having that kind of control over how often I can sample, but nothing beats seeing lemurs in Madagascar. just nothing beats seeing them in the wild, and the like hours you'll spend like staring at the forest floor trying to tell like fecal pellet from leaf matter. There's just nothing like that. It's a really wonderful experience.
How many times have you been able to go?
I think 7.
Amazing.
Oh, wow.
Yeah.
Wow.
So my wife is also a lemur biologist, and she's been working in Madagascar for much longer. And so I've gotten to really learn from her and get to hang out on a lot of her field missions. But the longest we spent together in Madagascar was, I think, 6 months at one time. So yeah, so these are
that's
dwarf lemurs.
That's picking the right field.
And the right wife.
You could be,
It is.
yeah, because you could study like an Arctic thing or a harsh desert environment or like something where it's like, oh, I've got to go into the field again. Oh, I've got to wear all this or keep drinking water all day. But you picked like one of the most beautiful places on earth. That's fantastic.
Well, it's not without challenge. And so I think in hindsight, we tend to romanticize these experiences like, that time in the field was so nice. Madagascar is like a real challenge, especially in the rainforest. It is steep. It is full of leeches. It is like, I fall a lot. I rip my clothes all the time. Everybody laughs at me, including myself. Yeah. And it can be a real challenge just to hike into some of these areas to even see the lemurs. So yeah, I like run a lot here, not because I enjoy it, but because I just don't want to be that embarrassed in the field ever again as I have been. So a lot of folks work in the dry forest because it's a lot flatter and a lot easier, although it's like incredibly hot and dry there. So yeah, it's beautiful and wonderful, but it is not without challenge. And especially if you're someone like Marina Blanco, my wife, she works on the nocturnal animals, and tracking those guys is a whole other like drama. Yeah. Just imagine trying to walk a mouse lemur.
You tap out, you're done with a day, tap out, she goes out for the night.
Exactly. I'm like, see you later. So yeah, I mean, trying to follow a mouse lemur around, it's weighs 60 grams in the dark, exactly. In the rainforest, it's pouring, and you're sort of hiking up hills. And yeah, I'll stick with the diurnals and the cathemerals.
Yeah.
That's a little bit better. That's another interesting aspect of ecological research that I've always been fascinated by is the bias of where researchers go and when they go and how that affects the data that we collect.
Yeah, and I think, yes, hugely yes. And for the microbiome field, one of the things that excites me is that we might, by using fecal samples, which we can collect non-invasively and from anything that poops, which is basically everything, we can actually level that playing field in terms of who we're studying. So a lot of the problem in things like characterizing what animals are eating means that you have to rely on animals that permit you watching them. And so a lot of our information about animal diets comes from well-habituated populations that have been studied for a really long time that often live in well-protected areas. And so we don't have nearly as good of an idea of what some of these sort of more at-risk populations might be doing or might be eating because they're scared of people. And so something like microbiome science, where we could use feces to actually reconstruct diets, either using microbiome sequencing or other types of sequencing, would enable us to really study more animals and sort of get rid of some of that bias that has been traditional.
What kind of outlook is there currently, considering climate change and deforestation and other human impacts on the environment and lemurs?
Yeah. So this really sort of sobering thought about climate change is that the future of lemurs in Madagascar is so dire that climate change might not actually become the principal threat. Some of these species are projected to potentially go extirpated in terms of populations or extinct in like really the sort of near future. And so a lot of us are much more focused on deforestation rather than sort of the less long-term but less immediate effects of climate change. So deforestation is a really big problem in Madagascar. Lemurs obviously need the forest to live. And so, yeah, it's pretty scary. We just recently, I just was able to attend the IUCN red listing workshop for lemurs. We just recharacterized the threat assessment for all 110 107, now 108 species of lemur, and so many species were uplisted to a higher threat category based on burning that's been happening, particularly in the West. So yeah, it's a scary time to be, I think, a lemur researcher, but it's also exciting to sit at that interface of where research-based conservation could actually come into play, to think about how we can use research tools to inform conservation action. Is sort of an exciting place to be.
Yeah, how you can go in and collaborate with the local communities to find ways to enact conservation, to find ways to make it say, this is in your best interest too. How can we work together?
And I think even not beyond local communities, but in addition to working with local communities, what gets me really sort of hopeful is being able to work with Malagasy scientists and see the enthusiasm for conservation and for science among my Malagasy colleagues, and being able to have all of our perspectives as part of a team is a really exciting place to be. So part of the reason I love working in Madagascar is getting to share that science with our Malagasy colleagues.
That's wonderful. Is there anything that you think people really need to know about lemurs?
I think getting to appreciate the diversity of species is really something that not a lot of people think about immediately. A lot of people think of like ring-tailed lemurs or rough lemurs, because it turns out that actually we have not that many species that are living in captivity. And so if you really start to look at all the species that live in Madagascar, you're going to see a wealth of diversity that you wouldn't get to see just by going to your local zoo. But definitely go visit and support your local AZA accredited zoo. But just getting familiar with all of these species that you'll never see unless you go to Madagascar. So things like sportive lemurs and woolly lemurs and the injury, all of these different types of dwarf lemur, fork-marked lemurs, the hairy-eared dwarf lemur. There are all these species that you wouldn't know about. So make sure you really appreciate the diversity that's out there.
And the dwarf lemurs, they're really interesting because they hibernate.
Yes.
Right?
They're the only primates that hibernate obligately. So every dwarf lemur of every species and every habitat hibernates every year.
Have to.
They have to.
Genes say sleep.
Well, so that's an interesting question, because in captivity, especially in the earlier years before they realized that they were hibernating in the wild, they wouldn't necessarily have the environmental conditions that would promote hibernation. So it's definitely a combination of your genetic machinery, but also the environmental conditions that trigger hibernation. And I should probably tell you to let Marina talk about this at some point, because this is really her area of expertise. So I can sort of be a bad mouthpiece for hibernation in dwarf lemurs. But so I think it was known for like a long time that these guys would go through seasonal fattening and seasonal weight loss, and that they would become sort of sluggish, but that you need to have the right environmental conditions at the right time to really trigger them to go into hibernation. But in Madagascar, they all hibernate.
Have you collaborated at all on the microbiome of the dwarf lemurs? And
we are doing that right now. And actually, we were going to submit a sequencing run before the coronavirus pandemic inhibited us from doing lab work. But we've been collecting samples from active and hibernating dwarf lemurs, both here at the Duke Lemur Center and also their wild counterparts in Madagascar, because there was this fascinating paper that came out in bears who also hibernate. And what the researchers did was they collected poop from active and hibernating bears. And then they took that poop and they showed that there were microbiome differences, but they transplanted those fecal samples into germ-free mice. So they gave mice the microbiome of an active bear versus a hibernating bear. Yes. And the mice that had the active bear microbiomes actually got fatter and gained more weight on the same diet than did the mice that had the hibernating bear microbiome. So the conclusion of this paper was that the microbiome is part of the machinery that helps you get ready for hibernation, helps you deposit fat in order to be able to hibernate. And this, for anyone that studied hibernating animals, was like, oh my God, I wonder if this is what's happening. So there are also groups studying squirrels and on all sorts of Arctic hibernators as well. So we are working on it.
Oh, I can't wait to hear the results of that. I hope that let's get the labs back up again, everybody. Slowly but surely, things will go back to normal eventually.
Especially a sequencing lab. You could send one person to go work with some liquid handlers. That's all they really need. And then they can be fine. PCR machine. That's it. That's one person can go into the lab and take care of all that. That person, your lab.
luckily, I don't actually do a lot of my own lab work. I send it to a national lab where they are much better at it than I would be. So I do the first preliminary steps, and then I ship everything off and rely on experts to do a lot of that PCR prep.
Yeah.
My
Sequencing is a very lonely job. They should have that should be going constantly. There's no need to shut that down.
Yeah, when I entered graduate school, my PhD advisor said to me, like, you have to figure out how you want to spend your time because you can't be good at everything. Do you want to be a field rat or a lab rat? And where do you want to focus your energy? And I was like, I want to be a field rat. So
Yeah, it's good to know the lab skills so that you know them, but yeah, it's hard to do all the things. It's good to, yes. Doing all the things. It's always difficult. I don't want to keep you too late because I know it is getting late there.
It's tomorrow here.
It's tomorrow already.
Oh, my
Way
goodness.
ahead of us.
It is tomorrow.
Yes. So where can people find you and find out more about the Duke Lemur Center as well?
So you can check out our website, lemur.duke.edu. You can also just look up the Duke Lemur Center on Facebook, on Instagram, on Twitter, and on YouTube. There's a ton of information and a ton of content out there. You can follow me on Instagram at Lemur Scientist, but definitely check out the Duke Lemur Center's Facebook, especially. They post like a ton of things like baby announcements and research updates and things like that. And there's a lot of amazing photographs and information there.
I'm going to have to do that.
Yeah, that'll be like the rest of your night.
Can we come get a tour once the world's back to normal?
Yes, once the tours are back open, you can definitely come for a tour. My stats might be a bit rusty, but I think it's usually like 30,000 visitors a year come to tour the Lemur Center. So we do have a full comprehensive education program that runs, and we're moving all of that to virtual right now. So there's a lot of virtual content that's also posted on our social media. Yeah, you can hear me rambling a lot.
And I understand that will be somebody wandering around in a full body lemur costume.
Yep. Yeah, we have a full lemur mascot that you do see around campus occasionally. Yeah. I luckily do not have to wear that. I'm very short, I'm not good.
Yeah, but that's too bad because I would have because then the lemur could have been busting out the ballet moves.
Right.
They do call the Shafox dancing lemurs.
Yeah.
Have you done Dance Your PhD?
I considered it, and then I realized that was a rabbit hole from which I would never return, and that I would spend a lot of time like choreographing it, and that I just let it be.
Well, coronavirus.
Maybe I can dance my postdoc.
Dance your postdoc, exactly. I would love to see it, the dancing Shafox. Thank you so much for joining us tonight. It has just been wonderful talking with you about lemurs.
Oh, really thanks for having me.
Yeah,
it's been fun.
Thank you. It has been fun. I hope you have a wonderful rest of your week and have a great night.
Bye, everybody. Thank you.
You are listening to This Week in Science. Thank you for listening to TWIS. You are the reason that we're able to do what we do every week, bringing you up-to-date and down-to-earth news and science views. That's right. And with your help, maybe, just maybe, we can continue to bring a more sane, maybe not completely sane, but at least a more sane perspective to a world full of misinformation. Head over to twis.org right now. Click on the Patreon link and choose your level of support. Be a part of bringing sanity and science to more people.
And we're back. You're listening to This Week in Science.
Yes, we are. Oh my goodness. Time for a COVID update. I do like this COVID music. Yes. Yeah. According to the Johns Hopkins COVID trackers, the U.S. remains number 7 in terms of per capita daily incidence and number 1 in terms of total daily incidence. India is poised to surpass the United States should we begin to decline a little bit more. Also, the World Health Organization data suggests a global plateau for daily incidence is emerging, which that could be potentially good news. Hopefully, we are plateauing a bit for this wave. And now, in the midst of Shark Week, we find ourselves searching for that evil lurking just beyond sight and wondering if it's safe to go back in the water. I mean, really, whether or not we can send the kids back to school. Yes. Well, although they don't present with symptoms as often as adults, there's a study out looking at the viral load in various age groups with mild to moderate symptoms, not the most severe symptoms, but the ones that don't really show much symptoms. And they found that young kids often have as much, if not more, virus than adults. And this doesn't say anything about their ability to actually transmit the virus now, but they have a lot of virus. They have all the virus. They have lots of particles in there, so... Oh. Additionally, a separate study found that 10 to 20-year-olds transmit virus as readily as adults. So this picture of kids as a viral petri dish kind of still holds. School not going to be this virus-free place if it happens. Also, 2 studies modeled different scenarios for returning kids to school. One in the Lancet found that in the United Kingdom, a substantial proportion of the symptomatic population would have to be tested regularly, and up to like 80% of the population would have to be tested. And contact tracing and isolation would have to be implemented and strictly followed if the schools are to reopen and not trigger a second wave. The second study in JAMA looked at college-age students and concluded that all students would have to be rigorously tested every 2 days to ensure safe campus reopenings. And this would cost about $400.
But it takes more than 2 days to get your test results back sometimes. Right? Okay.
Yeah, so they looked at a variety in the JAMA models. The different scenarios they looked at had tests with different amounts of sensitivity and specificity, so differing amounts of false positives and false negatives. And they determined that you could have tests of up to like 70% sensitivity, but the specificity needed to be like 99%. So one of the 2 measures needed to be incredibly accurate, but you could go with kind of a rapid, less sensitive test that are more or less expensive. But still, students would have to be tested every 2 days, and the cost would end up being around $470 per student per semester, if you're looking at a cost of like $10 to maybe $20 per test. So that we're talking cheap, not the best tests, but yeah. And I'm just trying to imagine a college environment where unless it's a saliva test, you want to go in and get screened every 2 days.
Yeah, but wouldn't that put 30% of the students in isolation at all times if it's 70%? Like it's
probably.
That's terrible.
Yeah.
So this is the part I'm not hearing from a lot of these studies. It's a difficult thing to really test or model because you have teachers that are coming in contact with different cohorts of kids, especially if you're at the college level. And then you have whoever those students of any age are going home to. If they're in college, if they live in a dorm with 6 other people, those people aren't in the same classrooms. We don't know what they're doing, if they've been to a kegger or a store lately. But for the kids in school, what if those parents are nurses or doctors or paramedics or janitors at the school or what, just anything else. It's just these social circles.
Yeah. That's why everybody all at once has to pretend that they got it and isolate for 3 weeks. It's the answer without a vaccine. It's everybody for 3 weeks.
I just keep thinking about that study they did with the buffet, the fake buffet, where they had one person that had the COVID like paint on their hand or whatever, and everyone ended up with it, right? So if you have one kid whose parent touched them as they put on their jacket, and then they touch a desk, and then the teacher touches that desk, and then the teacher takes it home, and then the teacher sees other kids the next day.
But even then, if you were talking about gloves and hand sanitizer, people are breathing it into an atmosphere in which that nobody can escape. So it's the aerosolized ability.
There needs to be more conversation about teachers, not just like their ability to spread from one cohort of students to the other, but also the risk to teachers. I'm seeing a lot about the risk to students, but not a lot about the risk to teachers.
Yeah,
I think that's a very fair point. There is a website that Marshall sent me, COVID-19 risk.biosci.gatech.edu, which allows you to dig into different counties in the United States to be able to assess the risk of going to events with certain numbers of people. So based on the case incidence of COVID-19 in your area or areas where you know people or maybe traveling, you can zoom in to the map to find locations. So for instance, if I click Multnomah County, Oregon, the current risk level is 68% that there will be at least one COVID-19 positive individual present at an event within the county of up to 100 people. And you can change the size of the event to be able to dig into different levels of risk. And so it's a pretty cool map to be able to start judging what risk you feel you're willing to put yourself in, right? How risky is something? There are some counties. So, for instance, Columbia County, Florida, for an event of 25 people, there is a 99% chance that at least one COVID-19 positive person will be at an event. So, based on the data, and this is based on seroprevalence data. So, they assume that there are 10 times more cases than are being reported. So, the rate may be lower where there's more testing availability. So it's not an exact estimate, but it's still very interesting to be able to kind of put into numbers, there are this many cases in my county, there are this many deaths. What does that mean? Or the rate is increasing of incidents. What does that actually mean? What is my personal risk? And so
that's very interesting.
Yeah. So
there was a conversation in Congress where a congressperson who is I'm not a fan of kept pressing Fauci on whether or not the protesters should have been arrested for being out there. The thing that Fauci didn't say that I think I'd point out is that we didn't see any protest related spikes, and part of what you'll also notice is those people are wearing masks. I mean, when you look at the protest footage, people are wearing masks and they're outdoors, which are two things that massively reduce risk factors. Keep wearing masks. Just wear the masks. If you have to go out, make sure you've got your mask on whenever you're interacting around humans.
Yeah.
and be outdoors whenever you can. City of Davis did something interesting along this lines. They did kind of reopen a lot of the bars and restaurants and that sort of thing. But all of Kiki, you'll be familiar with this, G Street. They got rid of the parking on G Street and built platforms where the parking was. So all these restaurants and bars, they got, instead of having their indoor seating, they have now taken over the street side where all the parking was, and put these platforms and have, outdoor chairs set at good distances. Is one of the things that they've done to combat this, because being able to space out and being outdoors are two massively reducing things. However, I'll still point out, if anybody has been downwind of somebody who is smoking a cigarette who's 40 feet away, this is how that was something that was in their lungs that's now out of their lungs. Okay, that's how easy it is for something to carry to where you are far away too. So you still need to have your mask on even if you're outdoors. But the combination of those two things massively reduce. It's the indoor and the non-mask wearing situations that are extremely dangerous.
And so the being outdoors, the physically distancing from other people, wearing masks, these are all social distancing measures. These are all things that we're doing to reduce the spread of this virus. And so the ability of businesses to reopen more normally, of schools to reopen, all of these things are contingent upon people continuing to follow the social distancing measures. We will not be able to open schools. We will not be able to get back to anything closer to normal unless people continue to wear a mask, physically distance, and try and stay outside, really, at this point. Winter is going to be interesting. That's all I have to say about where that's going to go for the time being. But my last story for our COVID update segment is lurking in our immune systems. This is, again, my, this evil from Shark Week. No, not Shark Week. Nefarious. No, there's an ancient power that today is understood as a functional bridge between our innate and adaptive immune systems. It is this bridge between the responses from what we have since birth and then what we develop in our immune system, the adaptive immune system, as we encounter various pathogens, that allows an integrated host defense to pathogenic challenges. And while the proteins that comprise this, what's called complement, are nominally part of the innate immune system, they can be recruited by actions by antibodies. So the antibodies that get triggered can then recruit the complement proteins, and complement proteins then help boost the adaptive immune response. And according to a recent study, this is potentially how SARS-CoV-2 virus is taking advantage of our immune systems. A recent survey of viruses discovered that coronaviruses tend to be mimics, like their molecular structure is similar to complement and coagulation proteins. What have we seen a lot of? Weird coagulation issues and weird inflammation issues in COVID patients. Based on that, on these symptoms that we've seen, and also this survey that found this kind of mimicry within coronaviruses, another group of researchers did a study in which they hypothesized that people who have hyperactive complement systems would be more likely to have severe COVID-19. And so they published a study this week in Nature Medicine that reports that people with age-related macular degeneration who have overactive complement systems are at increased risk of developing severe COVID-19. Exactly what they hypothesized.
Wow.
And so the finding suggests that the drugs that we have that work to inhibit complement could have use in treating COVID-19.
Interesting.
Yeah, so this really interesting logical path that researchers have been following to kind of dig into what is going on in this complex interaction between virus and immune system. Yeah. Some very interesting stuff right there. Yeah, complement. And I honestly, why call it a complement? I mean, it's a complementary system, I guess. These namings. Oh, scientists. If you just tuned in, you are listening to This Week in Science. Want to help TWIS? Leave a positive review for us today on your favorite podcast platform. All right, Justin, tell me a story.
Okay, so this is a drug candidate that has been developed by Salk researchers that was previously shown to slow aging in brain cells. By itself, pretty awesome. But now they've successfully reversed memory loss in a mouse model where the mouse were engineered to have inherited Alzheimer's disease. So this research, which was published online in the journal Redox Biology, also revealed that the drug, which is called CMS 121 at this point, works by changing how the brain metabolizes lipids. This is quoting voice of Pamela Mayer, who's the senior staff scientist at the lab of Salk professor David Schubert, who is also the senior author of this paper. This was a more rigorous test of how well this compound would work in a therapeutic setting than our previous studies on it. Based on the success of this study, we're now beginning to pursue clinical trials. So this is a molecule that is reversing memory loss. So basically what they did is they had, these were 9-month-old mice, which they claim is or use as a model of equivalency to middle-aged people. And these mice are beginning to show some deterioration, some memory problems. And they kind of liken it to the point when somebody might go in to do some cognitive testing because they've been having like an increased cognitive problems in their life. Maybe they know they have Alzheimer's in the family, maybe they don't. But it's enough for somebody to maybe go to the doctor to get themselves checked out. So that's when they began the treatment at this 9-month point. It lasted 3 months. So the now 12-month-old mice, both treated and they had untreated ones, were given a battery of memory and behavior tests. In both types of tests, mice with the Alzheimer's-like disease that had received the drug performed equally well as the healthy control group animals, while the untreated mice progressed in their disease poor performance on the test. That's pretty significant. This has not been an easy disease to treat, let alone there have been plenty of things that have attempted to just sort of hold where you are, but this one was doing a reversal. This one removed cognitive impairment from mice that were already experiencing it. So in this, they discovered that when it came to the levels of lipids, fatty molecules that play some key roles in cells throughout the body, mice with the disease had several differences compared to healthy mice and those who had been treated with the drug. In particular, researchers pinpointed differences in something known as lipid preoxidation, which is the degradation of lipids that produce free radical molecules that can then go on to cause cell damage. Mice with the Alzheimer's-like disease had higher levels of lipid preoxidation than either healthy mice or those that had been treated with the CMS 121. Quoting voice again, this is a postdoctoral fellow, James Attes, who's the first author of the new paper. That not only confirmed that lipid preoxidation is altered in Alzheimer's, but that this drug is actually normalizing those changes. They went on to also show that the drug lowers levels of a lipid-producing molecule called fatty acid synthase, FASN, which in turn lowered levels of lipid preoxidation itself. When the group analyzed the levels of FASN in brain samples from human patients who had died of Alzheimer's, they found patients had higher amounts of the fatty acid synthase protein than similarly aged control groups who were cognitively healthy, which suggests this is why we
That's a
have
big
mouse
role.
models. Which suggests that the fasten could be used as a drug for treating Alzheimer's disease. Other groups. While the grouping is pursuing the clinical trials, they hope other researchers will explore additional compounds that may also be treated by attacking fasten and lipid preoxidation.
Right. So they've got a drug. They've got targets, and it works in mice. So fingers crossed, it actually works in people.
Yeah, and I like that. This is Mayo again. She said there has been a big struggle in the field right now to find targets to even go after. So identifying a new target in an unbiased way like this is really exciting and opens lots of doors.
Yeah.
And it's one thing to open a door and have a path to a potential, as we usually hear in these early stage attempts to tackle a disease. But extremely exciting to have already had at least a mouse model example of it operating, having chased down from looking at other patients, having chased down where you think you might be a good target, and having it work is a little bit further down the path than often we're even talking about in the treatment of these diseases. So. Beyond a correlation, we have a molecule that seems to have impacted positively, and now having, what they're basically saying too is, hey, we did something that kind of worked here. Everybody else should steal this idea right now and attack here and see if you can do it better than we can, because we just want to get this disease off the plate. We're not
Let's fix
trying
it.
to corner. Let's fix it. Let's get this done.
Fix it.
Yeah.
Yeah. That's cool. Speaking of fixing things, there's a study looking at multiple sclerosis. Researchers say they have found the Holy Grail. And now whenever, that's sensationalist.
It's a buzzword for sure.
It's a buzzword. It's sensational speaking.
Somebody recently asked me what the equivalent of that is without having a religious aspect to it. What would be the most equivalent to finding the Holy Grail? And the one I came up with was finding the weapons of mass destruction. You're looking for something that might not actually exist, and you went to a war in the Middle East to find it. That's like
Holy Grail is the thing. It's the thing.
It's fix everything. The common cold would be a Holy Grail.
Yeah.
I still say it's starting a war in the Middle East to find something mythical. But okay, fine.
Okay. Apples, oranges.
They're the same thing. They're both round and edible. They're very similar. They both have the peel. They both grow on a tree. They're not that different. Researchers at the University of Edinburgh
worked on finding a way to help protect nerves in multiple sclerosis. In multiple sclerosis, there's a process called demyelination, in which the myelin sheath around the axons of nerves is damaged. And when that damage happens, it affects nerve transmission, makes it less efficient. And then also the axons then shrink and wither away. They basically become less and less and less able to function once that myelin sheath is gone. So the researchers were looking for mechanisms within the body that of people who don't have MS, the cells around neurons that work to protect and to support myelin sheaths. And so they have found something that they call ARMD. And ARMD is what they are calling axonal response of mitochondria to demyelination. What happens is in normal situations, when myelin is damaged and no multiple sclerosis is present, mitochondria are activated to travel to that area of damage. Mitochondria, being the energy powerhouse of the cell, then are able to energetically support the processes of repair and support the axon of the nerve that it's that glial myelin is a glial cell, and that the myelin is protecting. And so this is something that happens regularly, but when multiple sclerosis is in play, it's not enough. The ARMD, the armed, can't act enough to maintain the axons. The deterioration just continues to occur. And so they thought, what if we could enhance this axonal response of mitochondria to demyelination? And so they targeted mitochondrial biogenesis, mitochondrial transport, and they ended up finding a drug that is a commonly used, readily available diabetes drug that does this. And in their mouse model that they studied, that they tried all this out in, they were able to show reversal of demyelination in a mouse model of multiple sclerosis when given this diabetes drug. So it is one of the first times that a drug and potentially a manner of protecting the nerves has been developed for multiple sclerosis. And so similar to what you were talking about with the Alzheimer's study, where this is a target, this is now a mechanistic process within the myelin that can be targeted by, say, this drug that they found, or are there other better ways to activate those mitochondria to support? Yeah. So this is a couple of really interesting discoveries for some very big diseases that affect a lot of people. So I like to think, we were talking about COVID, where earlier in the COVID update, it's a little depressing, but there's some hope out there. People are still working on some really big solutions. So, but if you are trying to maybe someday hope that your brain will work like, did you see that movie Limitless? Where there's a drug and you could take a drug and your brain, its capacity was limitless. You could
Well, it was all based on the idea that that's not really true, right? That you only use like 10% of your brain or something.
Right. We use all of our brain, but we don't necessarily use every bit of it all at the same time. And this study that was just released out of the University College London was looking at the energy demands of the brain, and they're trying to figure out how attention, when you're focusing on something, and because you're focusing on something, you are not able to pay attention to other things, how focusing your attention kind of limits your ability to pay attention to everything.
Well, maybe for you.
I can multitask pretty darn well. But if I'm reading something, I sometimes can't hear. Somebody can be talking to me, and I don't even know that they're talking to me, because reading shuts everything else out. Yeah. Whereas I can carry on a conversation audibly with like 3 people and be listening to the fourth tape, not a problem. But if I'm reading, it just becomes tunnel vision.
Yeah, and so why does this happen? Why aren't we able to maximize processing, and what is the limit to this processing power of the brain? So there's study that was published in the Journal of Neuroscience. They were basically looking at this idea of how much energy it takes to run the human brain. And the senior author, Professor Nili Lavie, says, we know that the brain constantly uses around 20% of our metabolic energy, even while we rest our mind. And yet it's widely believed that this constant but limited supply of energy does not increase when there's more for our mind to process. So our mind is we're at this 20%. It's just, we're not taking any more energy from the body. That's just kind of what we've got. And so they wanted to see how the body was basically divvying up that energy during different situations. They used a new method of non-invasive optical imaging that they've developed at the University College London, in which they use a broadband near-infrared spectroscopy to measure oxidation levels of an enzyme that's involved in energy metabolism in the mitochondria in the brain. So most fMRI looks at blood oxygen levels, and it's this kind of it's not an exact measurement because it's a parameter that can be measured. That it's like, oh well, if oxygen's being used, then the brain's using energy,
Doing
right?
some kind of activity.
Right, some kind of activity, but we don't know what that oxygen really is an indicator of. And this is more specific in that it's actually measuring energy use in the mitochondria. And so using this technique and looking at the brains, the visual cortex of about 18 people, they gave them tasks where they increased the difficulty of the task and then had them pay attention to different things. So basically asked them to have singular focus and then tried to distract them and or tried to get them to multitask on things and look to see how the brain allocated energy. And they say that that really what they're finding is that the brain is going to fail to process some information because it just is taking that 20% and it goes, okay, you're focusing on reading right now. That is where the energy is going to be. And the energy that would normally go to other brain areas that are more for hearing or, I mean, they're only looking at the visual cortex in this particular instance, but looking at other aspects of visual processing, they found that the brain downregulated the amount of energy that was being used in those areas. So really less energy processing, less nerve activity in areas that are not doing the work. They say, the last quote from this article is, during recent months, we've heard from a lot of people who say they're feeling overwhelmed with constant news updates and new challenges to overcome. When your brain is at capacity, you are likely to fail to process some information. You might not even notice an important email come in because your child was speaking to you, or you might miss the oven timer go off because you received an unexpected work call. Our findings may explain these often frustrating experiences of inattentional blindness or deafness. But there's overload and there's a hard limit on our brain capacity for processing, according to this study.
I like the COVID lens on that because I think there's a lot of conversation about the emotional toll that all of this takes. And like, come on, it's a pandemic. Like, people are emotionally strained. We are physically strained in our brain, it turns out.
Yeah.
Our brain can only handle so much.
Yeah. And that's something that, potentially could be taken into account for, how much work people are capable of, how many tasks people are considered capable of, and as work-life balance is struggled to get to find, maybe workplaces can take this into account a little bit more.
Yeah, and this is we've covered this to some extent before, but this is also the effect of just being poor.
Right, because
just
there's a stress there.
There's a constant stress level, and if all of your mental activity is focused on the task of just having enough to survive for the other day, you don't really have sometimes times to make longer term plans or really focus on things like pandemics and global warming or any of the issues that you might be interested in if you didn't have to just concentrate so much of your time, energy, and stress on making ends meet.
Yep. Do you know what time it is?
What time is it?
I think it's time for Blair's Animal Corner.
With Blair.
She loves our creatures, great and small. Biped, milliped, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels. There's Animal Corner.
What you got, Blair?
Well, I have a story about pandas tonight, but it's about how they're the worst.
At what now?
Bad pandas.
They're just
What
the
are the
worst.
pandas the worst?
The worst
just the worst.
pandas.
Oh,
The
Yeah.
in every
worst.
category.
So let me explain. When you think about animal conservation, or when the average person thinks about animal conservation, what comes to mind? Of course, the panda. The panda is an extremely high-profile animal that lots of money and time and effort has gone to try to help save. And all in all, it's been pretty successful for the panda. They were removed from the International Union for the Conservation of Nature Endangered Species list. That's what our guest mentioned before, the IUCN Red List. In 2016, they're still vulnerable, but they were no longer listed as endangered. So that has worked. But critics like myself say, but we need to save more than just pandas. The response usually is that if you use a charismatic individual as the main kind of selling point for a conservation topic, then by saving that animal, you will inadvertently, or in some cases intentionally, also save other animals that live in that same habitat. And so that's been kind of the reasoning behind panda conservation for a long time, is even though, yes, they may be an evolutionary dead end and they always have an upset stomach and they're terrible at breeding and they get lost all the time, and I could go on, they still make a good focus for conservation because they are so charismatic, bring in money, and therefore help save other animals that live in bamboo forests. Well, as I mentioned, they're the worst. This is a new study looking at other animals that are endangered in panda habitat and how their population has done since panda conservation has begun. So new research has found that leopards, snow leopards, wolves, and dholes, they're also called Asian wild dogs, have almost disappeared from giant panda habitats since the 1960s. They looked at survey data from the 1950s to 1970s with information from almost 8,000 camera traps taken between 2008 and 2018. So they had their survey data, then they had their camera traps. And they found that leopards had disappeared from 81% of giant panda reserves, snow leopards from 38%, wolves from 77%, and dholes from 95%.
Pandas are really bad.
So they face a lot of the same problems as pandas. Pandas have problems with poaching, with wildlife trade, with habitat destruction. The predators face poachers, logging, and disease. And the key challenge seems to be that pandas have a home range of about 13 square kilometers. That's about 5 square miles. But the large carnivores I mentioned can roam over 100 square kilometers in habitat. So the fact that they save these small little islands for the pandas kind of don't do squat for these other animal species.
Yep.
So in the end, one of the lead researchers says they call for future conservation to see beyond a single species or animals with enormous charisma and instead focus on broader restoration of natural habitats, i.e. what I usually start yelling about on the show.
Yeah.
Save habitats, not animals.
But these also, I mean, the territory is all in China.
Mm-hmm.
I'm surprised that there's any wild just from the activities of Chinese. There were Chinese fishing boats off the Galapagos, like a fleet of them over the last couple weeks that had to get chased off. Like, who knows what they were going after? But yeah, if you picked one animal, you say, okay, this is going to be a controlled thing that's a publicity thing. We'll send it to zoos. We'll lend it out for $1 million a visit or whatever the thing is. Yeah, it's became a commodity, and I don't think it's ever been a conservation effort really for anything beyond a commercial enterprise.
So I think conservationists would disagree with you. There are huge projects working on establishing these protected spaces. There's breeding efforts where they release pandas in the wild. We've talked about studies on the show where they've done studies on animals that are born in captivity and if those pandas can thrive in the wild. And so there's a lot of good conservation work going on. My
For one
argument is
animal.
that it's for one animal and an
Yeah.
animal that I don't know if their niche is actually that important. They're not really good at surviving. Like, maybe let's try for somebody else that could do a better job.
Yeah.
Anyway,
Yeah,
it's
maybe try for something like the dole, which has lost 95%.
or just
and
try to save the bamboo forests, which is
Yeah, right. So I was
really
gonna say it's like save the biome itself.
Exactly.
This is the you say okay, here's this sort of biome that reaches from here to here. Here's the population of different animals that are there. We're just not going to let humans go there. And that's kind of
protect them all.
Yeah,
it's kind of the conservation you need.
I mean, and other animals have gone that way. If you look at orangutan conservation efforts, for example, it is 100% focused on saving rainforests.
Mm-hmm.
Okay.
It is not focused on saving orangutans. They use the orangutan as like the poster child.
Yeah.
But all of the efforts are focused on saving that habitat, which of course the rainforests are like the most biodiverse habitats on land on our planet. So good choice to save the rainforest. There's lots of animals that are going to benefit from that besides the orangutan. So it's a good reminder that that's where to go from here with animal conservation.
Thunder Beaver says charismatic is subjective, especially when it comes to the panda.
So it is and it isn't. I will say that some studies I've look at what makes an animal charismatic or cute. Usually charisma has to do with cuteness, right?
Mm-hmm.
And that's all based on infantilism. Do they look like babies? Do they have little baby eyes? Do they look fuzzy? So the more they look like a puppy, the better they're likely to do. And pandas definitely look kind of cuddly.
Mm-hmm.
So I would
and
not
the black
recommend.
around their eyes doesn't it takes away from the fact that their eyes are beady little eyes.
Makes them look big like a teddy bear. Yep.
Yeah.
Yep. Let's stop talking about pandas. Do you guys want to talk about bugs crawling out of frog butts? Because that's what I want to talk about.
Yes, I do. Now that you say it,
you know, predators prey. That's all life is about, right? The rat race. So prey can get away from predators a lot of ways. They can escape before they're eaten, but there are some that can escape afterwards. For example, some animals can survive the digestive systems of predators and are excreted in feces, and then they escape. But it takes a while. It's passive. I think we've talked about that on the show before too. There's like there's larvae that actually pass completely through before they're pooped out. But this is the first time we have seen a quick, active escape from the body of a predator after being eaten. This is from Kobe University, and researchers found that the aquatic beetle Regimbartia attenuata can actively escape from the frog Pelophylax nigromaculatus in their digestive
very well
system.
done. Well done.
Thank you.
Phonetics.
And where this comes from, how do you actively escape from the digestive system? Well, it is not enough to just crawl your way through. You must also get out at the end. Knock, knock, knock. So frogs don't have teeth. A lot of the time they don't kill things before they eat it. So usually the digestive enzymes are the ones that kill the prey so they can swallow stuff whole. And so these beetles have to not only scoot through quick enough to not get eaten up by digestive enzymes, but they also have to be able to get out the last leg of the journey, as you might say. So the sphincter muscle on the frog is the pressure keeps it closed. So the tiny beetles actually have to encourage the frog to open it. So they stimulate the frog's gut to promote excretion. This was true in this frog species and 4 other frog species that they tested this on in a lab.
So how do they stimulate it to promote digestion? I mean, is it like
Good
poke,
question.
poke, digest, poke, 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 or is it chemical stimulation? Like, what do they do?
I do not believe they know yet.
Or is it
what
just
they do
like-
know
or
is
is
that
it
if
just-
you chopped up the beetle and throw it into their body, it would take them over
Longer.
24 hours to poop it out.
Yeah.
But
Yeah.
if
ahead.
they were alive when they were swallowed, it would take about 4 hours. It would take way
Wow.
less.
So the beetle itself being present at the back door,
Yes.
at the back exit, would maybe be enough for the frog to be like, oh, it's not my usual time, but I guess I've got another one.
Yeah.
Here we go. Let me
Mhm.
just take care of some more business. I mean, I wouldn't that
Mhm.
seem logical that it's just, it seems like
Yeah.
it's ready again.
Yeah. Oh
to
no,
go.
that's definitely what it is. The frog. It's not like the frog's like, "What's happening to me?" No, they're going to poop for sure. That's part of the deal. But how? Yeah. How exactly are they doing it? How is it encouraging this to happen? Oh, it's about 6 hours. It happens about 6 hours after they're eaten. So a quarter of the time.
Quarter of the time, yeah.
Yeah.
Amazing.
And every single one that was excreted within those 6 hours, alive and active.
So, okay, so many questions and so many thoughts here. Mostly, I mean, this is just taking me back to so many cartoons growing up where main characters end up in the gut of the whale, or, I think there was a Schoolhouse Rock that talked about the digestive system, and they're inside the digestive system. You end up in there, and there's always this idea, fiction though, that you could get out.
Mm-hmm.
But it's not
Seems like you could.
fiction. It is real.
Yeah. So this is my question. The frog's not getting any nutrients from that beetle because it's not
No.
being broken down.
Why would it keep eating these beetles?
Why would it keep eating these beetles? This is the question.
Well, to be
It
fair,
just doesn't care. It's just, I want to
No.
eat anything.
No
to
harm,
be
no foul.
fair,
Maybe it cleans it out.
if you eat enough of them,
Screw.
it creates a line at the back, and then the ones at the back of the line end up getting digested.
Would they though? I don't know if they would.
I think so. I think
I think
eventually
they'd all
there's
just
too
conga
much
out of
traffic.
there. Make a nice conga line.
The gut escaping conga line.
Hey.
That's how they stimulate their way out.
Oh, yeah.
That's
I get it.
that's a survival technique that I don't think I've come across before.
Yeah. So
No.
this
Any
is
of our
totally
reports.
this is totally
We've
new.
never
This is-
talked about this before. Yeah.
No.
This is, as far as we know, the first study to report successful escape of prey insects from the vent of a predator. And
From the vent, that means
yeah.
the butt.
that means the butt, the cloaca, the all-in-one hole, as
Yes.
it were. And this is the first where we've seen that they promote excretion to escape. So this is a whole new strategy out there that needs to be studied. How do they do it? How do they promote excretion? Why
in
do the
fact, this is
frogs
the
still eat
first-
them?
And this is, so many questions, but this is the first time that they've seen it. So that in itself is, how common is this?
Mm-hmm. But the
Is it
fact
just
that 4 other frogs,
Yeah.
then 4 completely different species, also did this with this beetle. So we don't know.
Yeah, this beetle's just it's tough.
This
Mhm.
is
It's a
tough
tough
little
beetle. Eating me isn't gonna do nothing.
Don't
I'm
eat
not
the
worried.
beetle.
Here's some
love it.
teeth.
Yeah.
Oh my gosh, what a fun story to end the animal corner on. That was fantastic. We have one question for our This Week in Science questions. And we've talked about this a little bit kind of in passing, but the email we got said, hope you folks are staying safe over there, both physically and mentally. We haven't had a case in our state for a couple of weeks now, so fingers crossed. My question for TWIS is that for the last 6 months, there's been a large increase in personal hygiene. from social distancing to hand washing to the use of sanitizer, or just staying home. We've been exposed to less pathogens, and the drop in common diseases like influenza have been quite marked and measurable, and people are still having babies, maybe more than usual later in the year. Hey, lockdown was boring, right? As you know, the hygiene hypothesis is the idea that a decrease in exposure to microorganisms, particularly in children, leads to greater problems with allergies and immune diseases. I know time will tell, but do you think we will see more problems in the upcoming months and years? Or will this be a worldwide experiment to disprove the theory? Kurt Lansen. I don't know.
I don't know. I think for me, I still have a dog, so I'm still exposed to a lot.
And you're still going outside.
Yes.
And, yeah.
Yeah.
I think that is the big side of it is we're still going into the environment. You're still eating food. You're still getting groceries from various places. You are, if you have pets, maybe those pets are going outside, bringing things in. Perhaps you have children and they're in a small group, maybe not as large as they were. So the hygiene hypothesis, I think, really is more so like absolute cleanliness and also this real avoidance of germs that we've had as a society over years, with the idea that people in rural areas are, have fewer allergies. People who are exposed to cow pastures, for instance, have fewer allergies than people
who grew up with animals.
Right.
Or
Mhm.
yes.
Yeah, but there is something to be said about washing your hands way more, which we've learned really how often and how carefully we all washed our hands before, which is not great news if you're worried about disease, but maybe was good news if you're worried about this.
Yeah,
I think you're right, Kurt, though. I think it will be a good test. I think that you absolutely could see a potential of higher allergies, and also people getting a lot more cold.
Oh, he froze.
Getting cold because you're inside so much.
Yeah.
Long back to
Blah, 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 blah.
Yeah.
You froze for a second.
Yeah, we missed.
Wait, what just okay, first.
People being cold.
Without the kids going and getting the germ factory that they normally do in preschool and first and second grades and throughout that period. I think you could see people who haven't had as much exposure and have immune systems that are more likely to get colds. And I think those colds get a little tougher to deal with the older you get. I think there's something about that young immune system that can take on all these colds, always have, one line of snot coming out of one side of the nose to the other, and still be going about every activity as if nothing was happening.
And I think those kids
quite a test for that.
Yeah, and I think that, the kids not getting exposed to as many colds because, say, like Blair was talking about how with the school groups mixing, suddenly there'd be a new summer cold or something to that effect. But if you have fewer kids mixing in groups, what it means is, so kids are going to be exposed to fewer coronaviruses. Kids are going to be exposed to aren't going to be exposed to the flu this year, perhaps. Maybe that lack of exposure will lead to different vulnerabilities as they age.
Or maybe we'll kill some stuff. Maybe some stuff will peter out because it can't.
That's the other thing. There's some stuff that's not going to be spreading as much. But there's not that, amazingly, I don't think there's that much stuff that has a pathological effect on humans, really.
But there is evidence that being infected by some viruses does confer some amount of immunity to other viruses. And so even though we maybe have never been infected by SARS-CoV-2 before, but perhaps you've been infected by lots of other coronaviruses because you're around kids all the time. And so perhaps that makes you a little, maybe you get milder symptoms if you get SARS-CoV-2. Or,
Right.
there are even different varieties, different genera of viruses that confer.
Now, what will be kind of interesting also, though, is that with all of this staying at home, our microbiomes are probably becoming more unique to our immediate families.
More isolated to our home.
So we're not sharing microbiome out with the world so much, and we're creating our very own specific brand in the house between Blair and her dog and her main squeeze, and Kiki and the cats and the kid and her man, and me, it's just me.
And your rooster.
Doubling down. No, the rooster's gone.
Oh, recently.
Rooster's been gone for a long time, and nobody knows. I thought it was like, it's a farm. I figured I've been feeding the rooster. Was warning the rooster, getting kind of big. Don't want to be the biggest rooster on the farm at any point. But no, it wasn't. It turns out it's not the farmer, so we don't know. It might have been a coyote.
Yeah,
might have been
Yep.
Yeah.
Yep.
We've heard coyotes a few times out there.
Yeah, I think to the end of the questions, to Kurt's point there, like we are kind of about to see an entire generation of experimental conditions, not just for this, but also for the mental impact of this on all the kids who are losing potentially a year and a half of school and the social interactions therein. It's going to be a wild ride. It's going to be a very particular marker in our society that we will see.
But it's happened before, and for people who are saying things like, this is going to affect humanity in such a way, we are adaptable. If nothing else that we've talked about on this show so many times is the adaptability of humanity and our ability to weather very odd predicaments, to be able to take things in stride and continue. So, yes, there may be issues. There will be issues. We know that, but what they are, we don't necessarily know. We can try to make some predictions, but we've gone through these storms before. It will happen again, but we're going to be fine.
And also something to keep in mind: we have casually been had access to what we now would consider pretty harsh drugs that used to be very commonly prescribed by doctors, recreationally or prescription. We've had a lot of these markers throughout the eras outside of disease that we've done to ourselves, or chemicals in our everything that we've tried to get out now that we know that everything causes cancer, that we made stuff out of to cook with.
French fries cause cancer. What? Oh.
Yeah, it's bad, yeah, but it's not as bad if you cook it on your Teflon pan with the this is we've done a lot of mistakes that this compared to those probably. They used to painters used to retire from being house painters when the lead poisoning got too bad. Mm-hmm. This is we've had generations that
everything
had lead painted everything. Yep. And we survived. It's not the end of a thing. It's a marker. It's a hiccupy marker in terms I think of the schooling. By the time we're done, 1 million Americans being dead is not going to be any hiccup by any means. But in terms of having missed 6 months of school or 8 months of school or a year of school, it's the technology age anyway. Kids are probably learning more from a YouTube video than they do sitting around a class waiting for recess anyway. So if you put them in front of, if you have access to, you're doing that, Blair, but if you, it has to be the right one.
What they're watching.
Yeah. But if you put your kids in front of educational videos, they're probably learning more interesting stuff than they would be getting necessarily in a full day of school.
For 30 minutes. And then they're bored.
Yeah.
Well, yeah, because now that's the entire day of learning that they're used to getting at school anyway. Yeah. So, it's you've condensed it, and now they can go and play for the rest of the time.
I'm not against structured
learning, but
Yeah, back to the hygiene hypothesis. I really think that I think this is a blip, and I don't think that it is going to be as dramatic in terms of the hygiene hypothesis and that effect of leading to greater problems with immune systems and allergies. I think it'll just be, hey, you haven't been exposed to that coronavirus before, or haven't been exposed to the flu before, and so it'll be your immune system not knowing how to fight some things off. And so I think that is just more likely, which is just normal functioning of the immune system. But we will see, won't we?
Yeah.
If
I think it could get those higher ticks of allergies later on. I could see that.
Yeah, we'll see. If you have a question for us, send me an email, Kirsten@thisweekinscience.com. You can also leave us a message on our Facebook page, facebook.com/thisweekinscience. We'll do our best to answer. Whether we answer your question, that is to be debated. Thank you for enjoying the show. We have come to the end of another show. We've done it. Thank you to our guest tonight, Dr. Lydia Greene, for joining us this evening. And remember, you can find out more about Dr. Greene's research. Follow her on Instagram at Lemur Scientist. Shout outs to Fada for his help with social media and show notes and trying to herd the cat show that is putting together This Week in Science. Gord, thank you for manning the chat room. Identity4, thank you for recording the show. And I would like to thank our Patreon sponsors and the Burroughs Welcome Fund for their generous support. Thank you. Oh, it didn't change the page. I was going to do it, and I was doing other things, and then Give me 1 second. Apologies to all. You like my... It's my circus hold music. Okay, thank you to John Scioli, Erik Combs, Flying Out, Guillaume, John Lee, Ben Rothig, Ali Coffin, Maddie Perrin, Gaurav Sharma, Taylor P.S., Josiah Zahner, Mike Shoemaker, Sarah Forfar, Donald Mundis, Rodney Lewis, Stephen Albaran, John Ratnaswamy, Dave Friedel, Daryl Myshak, Stu Pollock, Andrew Swanson, Corinne Benton, Sky Luke, Paul Runovich, Ben Bignell, Kevin Reardon, Noodles Jack, Sarah Chavis, Paul, Jason Olds, Brian Carrington, Matt Bass, Joshua Fury, Sean and Nina, Sue Doster, John McKee, Greg Riley, Mark Hessenflode, Jean Tellier, Steve Leisman, Ken Hayes, Howard Tan, Christopher Rappin, Richard, Brendan Minish, Melazond, Johnny Gridley, Richard Porter, Christopher Dreyer, Mark Missaros, Artyom, Greg Briggs, John Atwood, Robert Rudy Garcia, Dave Wilkinson, Matt Sutter, Philip Shane, Kurt Larson, Craig Landon, Mountain Sloth, Jim Drapeau, Alex Wilson, Dave Neighbor, Matthew Litwin, Eric Knapp, E.O., Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul Disney, Patrick Pecoraro, Gary S., Ed Dyer, Tony Steele, Ulysses Adkins, Brian Condren, and Jason Roberts. Thank you for all of your support on Patreon. If you are interested in supporting us on Patreon, you can find information at patreon.com/thisweekinscience on next week's show.
We will be back Wednesday, 8 p.m., broadcasting live from our YouTube and Facebook channels and from twis.org/live.
Hey, do you want to listen to us as a podcast? Just search for This Week in Science wherever podcasts are found. If you enjoyed the show, get your friends to subscribe too. Maybe tell them that we talked about, beetles coming out of frogs. It'd be nice and fun.
For more information on anything you've heard here today, show notes and links to stories will be available on our website, www.twis.org. And you can also sign up for our newsletter.
That's right. Sent one out today, actually. You can contact us directly. Email Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or Blair at blairbaz@twis.org. Just be sure to put TWIS, TWIS, in the subject line so your email doesn't get spam filtered into oblivion.
You can also hit us up on the Twitter where we are at TWIS Science, at Dr. Kiki, at Jacksonfly, and at Blair's Menagerie. We love your feedback. If there's a topic you'd like us to cover or address, a suggestion for an interview, a haiku that comes to you tonight, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
It's all in your head.
This week in science. This week in science. This week in science. This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer and it shouldn't be news. That what I say may not represent your views. But I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. Jeopardy. And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 Got a laundry list of items I want to address. From stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought. And I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say. And if you've learned anything from the words that we've said, then please just remember it's all in your head. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.
Dun, 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 dun. We're in the after show. So Erik in Alaska says, fun fact, that circus song is called The Entry of the Gladiators. Fun fact. Tonight's show was not funded by the Juggalos, Thunder Beaver.
Rude.
No. I mean, maybe it was, actually. I don't know. I mean, aside from people's names, I don't know how they spend the rest of their lives other than supporting TWIS. So you know what? We may be supported by some Juggalos. I can't say these things. I don't know.
We are definitely not affiliated.
No, we're not.
That's
not
for sure.
affiliated. That's for sure. Yes. Beth Bennett says about the frog. To be fair, that frog's brain is pretty small. So the frog thinking about things. Yes.
I mean, it's all relative.
It's all relative. Gaurav Sharma, so sorry for throwing that vent escaping beetle story at you when you were eating.
Well, you should know better when we enter the animal corner.
Yeah. Hello, Wing of Tech. Hello, hello. Oh, my goodness, Gaurav. The Beirut explosion was that's horrific. I mean, I think we're all unfortunately a bit numbed because of coverage of wars around the world and various various fights and bombings and other things, but stuff that happens like what happened in Beirut this week it's shocking to everybody. It made its way around Twitter really fast. Did you hear about this, Justin? You're looking squinty eyebrowed.
Yeah, it was a couple tons of chemical that had the story I heard that was taken off an abandoned ship or something and stored there for 7 years, and then they also put in the warehouse fireworks and other things, and then
I think it was a fireworks factory's warehouse, and they had stored it there. There are a few different stories. One report I heard said that the fire started on a ship and then spread to the warehouse.
I didn't hear that.
Another, but because it was down at the docks, maybe that's why there was story about a ship at all. But yeah, the warehouse. There was the warehouse that had fireworks chemicals in it, and it did a big boom.
The way I heard it was two different things. Like partly it was storing fireworks, but they were unrelated to the 7 years of 2 tons of what was it? Something that's not supposedly that it's not an explosive by itself, but yeah, if you ignite it, I guess it becomes. And there was tons of this.
Highly.
Ammonium something or another. I'd have to go find a version of the story I read. But yeah, it had nothing to do with
ammonia.
This was.
I thought it was.
Ammonium something.
Nitrate.
Sulfate?
Probably
I don't
ammonium.
know. Okay.
Ammonium nitrate.
Yeah.
Yeah. Ammonium nitrate.
But
yeah, I wonder what it was. If it was ammonium
nitrate.
No, I'm kidding. I'm joking. Like Kiki's just like, no, but I actually know what it actually was.
Ammonium nitrate. Yeah.
But
yeah. So if
but like 2.5 tons of it that was just being stored for 7 years.
Yeah. Yeah. I've seen some interesting calculations based on the blast and how the diameter of the blast as the explosion occurred, and using that change the speed at which the diameter of the blast, or the blast radius, I guess, expanded. So there was one calculation that someone shared with me online that an engineer had attempted to calculate backwards to the equivalent amount of TNT based on that changing blast radius. So there's some interesting analyses that have been taking place, but it's amazing how fast people are like, oh, that was a nuke. There was a mushroom cloud. And it's just because there was a mushroom cloud doesn't mean that it was a nuke. It just means that it was a very big explosion. Yeah.
Oh,
Gary Lubert is saying
my number is way off. They say it was 2,750 tons.
Yes,
2,750 tons of ammonium nitrate that had been sitting in this warehouse for 6 years.
Yeah.
Without anybody's responsibility. Wow.
That's what warehouses are for, aren't they? Yeah. It's like, it's like those those
No.
storage spaces that you rent where you put like your old furniture that you can't bear getting rid of, but that you're never going to use again.
Yeah. There's supposed
Yeah.
to be regulations and things in place to prevent things like this from happening. But again, the version I heard was this was confiscated off of a ship and stored there. And then nobody's like property,
checked on it.
nobody's
Yeah.
responsibility, nobody really, other than don't touch it. Like, apparently for 6 years happened after that. So yeah, that's not cool.
Yeah.
But
Not
yeah, it
cool.
doesn't sound like any
a
kind
lot of people injured and dead and homeless now because their apartments have been destroyed. Yeah. What if your couch was carved out of C4?
Nothing.
Identity 4. Nothing. Don't you need to actually give C4 a stimulus to make it explode? I'm going to totally be on a watch list right now. Yeah.
But yeah, so without a government response, something that could have been handled with very few deaths spirals into something that has caused much destruction and many deaths. Not unlike what took place with COVID here over the last 6 months of inaction. So. There's people don't like red tape. We all get it. That's annoying. But having regulations and laws and responses that are based on science, and material handlers in this case should have been sent in to assess whether or not this was okay to have there.
Yeah. Good night, Fada. Yeah, it was very similar to the port of Tianjin that exploded in China. Yeah, very similar. Ugh. There have been a few of these kinds of explosions over the years, and they're always just shocking.
We had a aircraft carrier explode in San Diego. Texas, like every year, has some big factory or fertilizer depository or chemical something blows up and takes out. It's usually in rural Texas because that's what Texas is.
Yeah. Erik in Alaska is going back to the Halifax explosion, which was the largest man-made explosion before Hiroshima. Yeah. Another big one.
I don't even know what that is. What is that?
Just look it up. 1917 Halifax explosion. Ooh, Erik in Alaska says it's a munitions ship that blew up in Halifax Harbor. That was even before we had the big booms.
I'm sure everything is in boxes and tied down, I guess.
Yeah. Anyone have any good news from this week? Good things happen? Nobody's got anything good. Blair wasn't listening. She's like, "What? Hello?"
Just said goodbye to my EMS worker for the night before he goes back into the fray.
Yeah, how are things in San Francisco? I know that in California, rates are up.
They were okay for a while. For a while, I was like, "Any COVID last night?" He's like, "No." But now there's definitely some like every night. But it does seem like they're talking, like they're like getting ready for the storm. Like they're all bracing themselves for it to get really bad.
Hopefully it doesn't.
Yeah.
hopefully people are staying home and hopefully they're wearing masks and it doesn't get worse.
Well, now that like
Past the peak. Maybe.
summer's over, maybe people stop making dumb decisions. I don't know.
No, we still have Labor Day. Come on.
That's true. You're right. Labor Day is
It's a
going to be a whole
whole
thing.
nother holiday coming for people to make
Yeah.
dumb decisions. Come on.
You're right.
This is the year of living simply, the year of, practicing distance and keeping in touch with people in different ways. We can do this.
I mean, can we?
We can.
I can. You can. Justin
We
can.
are.
Most of our listeners probably can. But can we do this? I really hope so.
Yeah. Gaurav Sharma says it seems like we're getting better at treating COVID symptoms. So that's some decent news. Agreed. Gary Lubert says love is real. Agreed. Erik in Alaska says the weather's been nice. Agreed. These are all very good things. Thunder Beaver got a new mobile office back. Good job. Identity 4 pulled lots of onions out of the garden. If you like onions, that's great.
I love onions. I love to saute an onion.
Ooh, a brown bear. Erik in Alaska saw a brown bear at a safe distance last week.
That's a big boy.
I don't.
If you
see
weren't in
anything like
Alaska,
that.
Erik, if your name didn't say you were in Alaska, I'd be like, nah, you saw a brownish black bear. Because that's a big thing, is people say, I saw a brown bear once. I'm like, yeah, how big was it? Was it as big as your car? No? Okay, then
Nope.
you saw a brownish black bear. Yeah, that's yikes. Brown bears are no joke. They're so big.
I just want to stay away from bears. They're too big.
I got within like 5 feet of a coyote this past week. So it's
How was that?
it's darker in the mornings, but my alarm goes off at the same time. And I take Sadie out for a run. And so I went outside at like 5:50 in the morning. And
Heard the words.
She's like, I hear you.
sweetie, I'm here.
Heard the word walk.
And so I took her out to go to the bathroom and stuff, and I saw a dog just like trotting towards me. I was like, where's your owner? And then like as it got closer, I was like, nope, that's a coyote. And it was just kind of like minding its own business, just like walking from point A to point B. And I could have reached out and touched it. It was so close to me. Luckily, I went to a talk all about coyotes last year, and so I knew what to do. And so I like backed a little bit, started clapping, started shouting, just like, get out of here. And then since he didn't leave, like the coyote just was looking at me. I picked up a pine cone.
I don't care.
And I threw it at its feet. And then it like turned around and was like, all right. And then like started to trot away. And then at one point like sat down and scratched its ear and then got up and continued to trot away. Just like not giving a crap.
No. I think that's one thing that we're going to see. It's the impact of humanity that normally makes these animals fearful. They're getting bold. They're finding food. They don't have as much food because all of the trash and other things that maybe they depend on, or the rodents that feed on the trash that humanity produces and leaves out, it's not there. So they're getting bold.
Wing of Tech says, I suspect raccoons to be bathing in our pond with waterfall in our front yard. What do I do? Leave out towels? Very small towels.
Very small. Nice towels. They'll be like, oh, thank you very much. It was a good bath. It was lovely.
I don't think they're bathing in your pond.
Drinking the water? Pooping
I think
in
they're
it?
drinking the water.
They're probably drinking it.
Yeah. To my knowledge, raccoons are not super into taking a dip.
I think it's an interesting question, though.
Yeah.
Which animals do take baths?
Yeah.
I mean, they're so fluffy. They probably wouldn't want to. For virus to spill over, asks Gorov, does the human have to consume the animal, or even being a zookeeper can do it?
So consuming the virus doesn't really do much.
Yeah. Unless it's something that is a virus, not respiratory, but more systemic, and it's you're eating raw meat.
Right.
Maybe, but that's usually more often like
Come out here.
a parasite or something. But
So wasn't Ebola bushmeat,
Bushmeat.
blood-to-blood contact? Like
Mhm.
you're cutting up a butchering an animal basically, and you have a cut on your finger somewhere, and
Mhm.
that's how the virus could get in.
Yeah. And then viruses, the respiratory viruses, that's just going to be, going into a cave where a bunch of bats are and they're guano, and it's just aerosolized in the air and you breathe it in.
Or types of bird viruses
Mhm.
that you can get that way, or from rat feces.
Yeah.
it's,
Yeah, so it's not consuming. Not normally. Yeah, I think consuming, the one that I've really heard of, it's not a virus though. It would be the prion.
Mm-hmm.
When you consume deer meat that has the all I can think is kuru, but it's not kuru. It's the virus that causes Creutzfeldt-Jacobs. Creutzfeldt-Jacobs. That one. I think Blair's tired.
How can you tell? Because I went
getting
from
lower
standing
and
to
lower.
sitting.
The twisting
Yeah,
takes
as
a while.
previously mentioned, that 5:45 alarm comes real quick.
It comes early.
Yeah.
Yes. How's your work, Justin?
I'm not. I'm done. I'm done working. I'm now on a countdown to leaving the country.
Really?
Yeah. Hey, anybody know how phones work? I got a new phone. It's unlocked, and I moved my SIM card from my old phone to the new phone, and I don't know if my carrier settings aren't on the SIM card or
Wait,
what.
did you have it's Apple products?
Yes.
Okay, so do some Googling, put your SIM card back in the old phone, plug your
Did.
phone into your computer, and then you
Ooh.
have to start a process to transfer from one phone to the other.
So I transferred all the data. Got transferred. This just had me hold the phones next to each other. But
It's
that's
a
something
carrier
else.
settings. So you have to go to your carrier's website and say
Okay.
I'm activating a new phone, and then it'll be like, all right,
Oh.
now turn your old phone off. Okay, now plug in your new phone.
Okay, I get it. And then with the SIM card in the new one.
Yeah.
Okay.
You don't actually need to move your SIM card. In fact,
What?
they don't. My phone. I have an iPhone 8. It doesn't have a SIM card.
Oh.
So.
What? Yes, it does. Yes.
Nope.
Yeah, I have a XR now, and it has it.
Yeah.
I got my daughter an 8. It had it.
Well, then you don't need to transfer the SIM cards anymore. That's not a thing anymore.
Yes, you do. Yes, you absolutely do. Yes. I'm not taking your advice on that. In fact, I got this phone so I could have 2 SIM cards in it.
Then let me ask you this, Justin. Why did you ask?
Well, no, they're getting the carrier settings updated via computer from the website. That makes total sense.
It told me not to pull out my SIM card. It told me very specifically. It was like, nope, we're going to do it all.
That just does not make all of the sense.
Your SIM card is your carrier information and your contacts, and all those things will get transferred.
Really?
Yeah.
Then why do I have 2 SIM card slots on this one?
Probably for international travel, so you could put an international
exactly
carrier on
But then I don't need my regular one anymore? Is that what you're saying?
I'm saying that you don't need to physically pull your SIM card out of your old phone.
Find this information dubious. However, I will take your advice, and I think that makes sense. There should be
Although, actually, if you bought an unlocked phone, maybe you do.
yeah, it's not.
If you didn't buy it through Apple, you might need to. Or through your carrier, then
through
you
the
might
carrier.
need to.
Through the carrier makes sense. Yeah.
But I bought mine through Apple,
Okay.
and I didn't need to move my SIM card. I just did this like a couple months
So you
ago.
don't have a SIM card.
Or if I do, it came with one.
Have
in
you
it.
left the house with the phone and used it? Just asking.
You know, I'm
just
working
have-
full time offsite, right?
But is there Wi-Fi everywhere? Like these things are
Oh my
important.
god, Justin!
Justin. Blair is too tired.
The level of condescension is like thick.
Oh no,
No,
it's not condescension.
It's absolute dubiousness. Because like this is like what I thought the whole point of a SIM card was. It was
Yeah,
like
well,
that's how you
it's
make your phone
2020,
do
my
cellular.
dude.
I get that, but then why do they have like more slots now than they had before? If it's
I think
like the
that's so you can
obsolete
do
thing?
both. I think it's if you want to have like dual carriers, then you might need a SIM card in there.
Mhm.
But if you have a phone,
Or
it's
if you're going
basically just a
international.
computer, right?
Yeah.
That's like linked up to a network.
It does seem weird that the chip little chip I had in there from I don't know 10 years ago with my first iPhone is still necessary to put into
That's what I'm
this
saying. I
newer
don't
phone.
think it is. I don't think I have one anymore.
Eric Knapp says carriers are going to eSIMs
Yep.
and
There we
away
go.
from physical SIMs.
There we go. See,
Okay.
I'm not full of it.
I didn't say you were full of it. I was just like, then why is this a thing still? Why does like have like
You got to
It's
program
sort-
the computer, man.
Why are CD-ROMs still a thing?
I mean, yeah, why do their computers still have those? Like I
Yeah,
guess
that's
they
right.
don't.
I put
don't
the
have
ROM
one.
back in CD-ROM. I remember when they were called DVD-ROMs.
All right. No, I'm learning from you, Blair. I'm not trying to be condescending in the very least. I'm just like
Okay.
not getting it. Is all.
All right.
It's an okay boomer moment I'm having. Just
All right.
be gentle.
You're not a boomer.
You're not a boomer.
Saying Apple doesn't have SIM cards, but my Apple phone has 2 slots for SIM cards. So you
Yeah,
can't just
that's
tell
what
me
I'm
that.
saying. It's for international travel.
Okay,
It's so you could have an Italy SIM card and a Greece
Yeah.
SIM card.
yeah,
in
that's what I'm
there.
gonna do. I'm gonna have like all these different
Thank
phone
you.
numbers in one phone.
Okay, you guys, I'm gonna turn up my input on my audio because Identity 4 says that I'm still quieter than you guys, which is all the
Mhm.
time. Even though when I'm just recording into my audio recording software, I have perfect levels. They're perfect. But apparently I'm quieter than you both by about 6 decibels. So I'm going to turn it up right now. Just turn up the level and see if that works. Did that work? Am I
I think
now
that did.
even with,
I think.
or
you
am
did.
I louder than Blair and Justin?
You
I
sound
can
good.
mark
It
it.
still
Yeah, you
sounds very
sound good.
warm.
We absolutely hear you.
Sounds good. You don't sound overexposed or anything.
Not at
I
all.
I don't like to be overexposed. The sun, my skin, it doesn't go well together.
Mhm.
Identity 4, am I still 6 decibels? Yeah, everybody talk at the same time.
Blah,
No.
blah, 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 blah.
Now, Justin, you're being super quiet.
No, I'm not being quiet. Why did I do that?
I'm not being quiet. Why would I be quiet?
Identity board just says it's louder than it was.
Great. Great.
Yeah,
Okay.
it
I do think that is how knobs work.
I mean, I could turn up the aux. I can turn up the gain. I could turn up the level. There's a lot of knobs.
So many knobs. That's why I gave up on
The mixer.
the
You're like,
mixer
I'm
I
done.
had. It had like 100 knobs.
I made myself a little more gain. How's that? How's that gain? Oh, wait. Now you say now that's better, but now you're behind. Oh, did I make it too loud?
No, that's fine. I can hear you loud and clear anyway.
Why are you whispering?
Oh, now you're real hot, Kiki.
Okay,
Yeah,
I'm going
you're
to turn
right.
it back down again. Turn that gain back down. Justin, talk for real.
What do you mean? I'm fake talking.
I know you are.
No, I'm absolutely not.
When do you leave, Justin? Do you have a flight already?
Oh, yeah, I do. I need a calendar to figure it out, but let's see. What is today?
The 5th of
Less
August.
than 2 weeks.
Hmm,
The 17th.
that's great.
Mm-hmm.
Better balanced, better balanced, better balanced. Better balanced, better balanced, better balanced. Okay, so in about 2 weeks, week and a half, we're going to be dealing with Justin
On Denmark time.
on Denmark time.
Oh, yeah, I've been doing this show at, oh, gosh. What is it? 5 o'clock in the morning?
Yeah.
Yeah,
Until
early
time
riser.
changes help that out slightly
For a bit.
for a bit. Mm-hmm.
No,
Are you
it's
going
5
there?
o'clock in the morning. Sorry. It's 5 o'clock in the morning there is when I
Yeah.
would
Yeah.
do the show,
Yeah.
which means
You have to get up.
it's 8 o'clock here still. That's because you guys are in the same time zones. You're not
We don't
Mhm.
change.
changing.
No.
No, you'll stay the same. It'll be me that's operating in the morning. It'll be it'll be a morning show. Disclaimer, 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 everybody. It'll be like that.
It'll be you waking up at 4:55.
No, I'll be up at 4 because I got to prep for the show. I got to find my stories. Drinking coffee. And yeah, it'll be a completely different scene than this one here.
When I was in Israel, we
Sundays.
were still doing 7:30.
Mm-hmm.
And so for half the time I was there, I had to do it at 5:30. And then for half the time I was there, I had to do it at 4:30. So I woke up at 4.
Yep.
Great.
I remember those shows. You don't.
No, I don't. I really don't. Sadie, you're okay.
Oh my goodness.
He's just barking from the bedroom. He's like
Okay.
too lazy to get off the bed, but still like, hey, what's going on out there?
What's going on? What's going
Go back to bed.
Oh, actually, it's according to this, I can just dial a number. Maybe I don't even have to be connected to a computer.
Mm-hmm. Yeah, you could do that too.
What? What?
For
I can
his
try.
phone activation.
Oh, for your phone activation. Okay.
All right, my dog's roofing up
Yes.
over there. She's going roof, roof.
Because you said walk.
Eh, well.
Now the dog's like, well, we're
She
going.
needs to learn
We're
context clues. I'm sorry.
Learn. Learn, Sadie. Learn. All right, everybody. Friends, thank you for joining us for another episode of This Week in Science. Thank you for joining us and spending so much time with us this Wednesday evening, learning about lemurs and their poop, and learning about beetles that aren't poop but act like it, and all the other science fun.
Yeah.
Beetles crawling out of frog butts. You're welcome.
Incredible.
Oh, I want to go hug my cat too. I'm going to go hug my cat.
Incredible. Yeah.
Be careful, there's no beetles crawling out of his butt.
Ah, no.
Is it just frogs that eat this beetle? I'm curious.
We don't know. I don't think.
What
they've,
happens
like,
if
gotten that
Justin
far
eats
yet.
the beetle?
Or a
I
bird.
mean, I
What if a
assume
bird
they'd
swallows
crawl
the
their way
beetle?
out.
Yeah.
Knock at the door.
Excuse me, I need to leave, please.
Yeah.
Excuse me, would somebody please open the door? I'd like to go now.
Yep.
Okay, Blair, for next week, I
Yeah.
really, really want some bad panda art.
Bad panda. Okay.
Bad panda!
Bad panda.
Bad panda! I mean, seriously, I'm like thinking of about pandas with all sorts of social issues. No.
Say goodnight, Blair.
Night, Blair. Say goodnight, Justin.
Good night, Justin.
Good
Good night, Kiki.
Kiki.
Good night, everyone. Thank you. We will see you again next week. And in the meantime, we hope that we see you on the interwebs. Have a wonderful week.